80 students are together in one place, and even in the lunch and the other time, those are the time that you can express yourself, what you want to do. There is no need that you need to explain your poster and other things during that social time. Just pick up some ideas and talk to them. At least this 2 different, tea session that we have, and then one lunch we have. So I want to see the students to grab one of the experts in here and then just talk to them, anything you want, okay? Don't leave them to be together. So that is my humble request. Thank you.
We'll keep that in mind, sir. Thank you. So we'll begin the 3rd day today, which the focus is on human health. Session is on microbiomes and health. The session chairs for this session are Dr. Devika and Dr. Suresh Sudarshan. Dr. Devika is an assistant professor at Sastra University, where she has been teaching undergrad and postgrad students. She completed her PhD at Anna University, where she conducted an in silico investigation in regulatory mechanisms of endothelial nitric oxide synthase. Her research focuses on exploring probiotic signatures using machine learning-driven genomics and metabolic modeling to understand their health effects. Dr. Suresh Sudarshan is a distinguished bioprocess engineer specializing in microbial physiology and systems biology. He earned his PhD in biochemical engineering from the Technical University of Dortmund, Germany, and he has joined Novo Nordisk Foundation Center for Biosustainability at DTU. He's now a senior program manager for the High Density Physiology program. His expertise encompasses biotechnology research development, microbial physiology, and dynamic modeling. Welcome.
Thanks, Mazia. So very good morning to all. So today we are in the 3rd day of Symposium, and the session number is 7, and the theme of the Symposium is microbiome and health. So I'm very happy to introduce our first speaker, Dr. Gaurav Sharma. He's an assistant professor in the Department of Biotechnology at the IIT Hyderabad. And earlier to this, he was working as a DST Inspire faculty at the IBSE Institute, Bengaluru, and then he's done his PhD in microbial genomics from CSIR Institute, Chandigarh, and his research mainly focuses on microbiomes, microbial genomics, and evolution, with a particular focus on understanding the microbial systems using advanced genomics and transcriptomics approaches. And he's also recognized for his contributions to science-winning awards like AMI Young Scientist Award and the BioClues Innovation Award. So over to you, sir. So the title of his talk is Tracing the Evolutionary Threads of Myxobacterial Flagella.
Can you hear me now? Great. Okay, so thank you very much for the introduction. So today I will be talking about tracing the evolutionary threads of Myxobacterial flagella. Before we start, as the tradition is kind of going on, I would like to thank Karthik and Karthik in a different style. So we do a lot of homology analysis. So I thought of that, okay, we have 2 Kartiks here. Let's do some homology. And we found out they are 63% identical, as you can see. We also try to make some dot plots just to see, you can see that. Okay, so thank you very much, Karthik, and it's a great opportunity for us to meet a wonderful panel of speakers here, and I'm really enjoying and looking forward to coming back to this conference again and again with more and more students from our lab. This time we have only 2, but next time I'm sure we will have more. Okay, so my lab work a lot on sequence, structure, and function along with evolution. This is the main direction where we are trying to do. We do a lot of alignments, structural alignment, functional understanding of each system, and try to see from the lens of evolution. There are several focus areas which I have been working in the last few years. The main one stays microbial genomics and evolution, but we have also started doing medicinal plant genomics and metagenomics to understand the plant microbe interaction, and we are also collaborating and also making some prediction servers. I will be very happy to chat to you about several of them. But yeah, feel free to go to our website and find out more about these few servers, especially this Google Scholar Lens, which is kind of a very new feature which we have added. So today I will be talking about myxobacterial genomics, and this work has been done by Utkarsha. She has a poster here. We have another poster by Shravani, who is working on family Vibrionaceae, trying to understand how core genes move from one chromosome to another, a very fascinating study which we are coming up with. Okay, so let's go ahead with the main talk here. So we work with Myxo. Myxo, if you guys don't know, they are social microbes. They are actually a very fascinating group of microbes who stays together, do a lot of things together. So obviously everyone will ask why. But so let's go. I will tell you like 3-4 things about these organisms which will allow you to appreciate this particular model organism. So they are aerobic organisms, most of them, and they come within delta proteobacteria. And if you know, delta proteobacteria are mostly anaerobes. So the first question is that how this aerobic respiration evolved within these organisms, and we have found several connecting links also, which I will quickly discuss between these two organisms. Second is that they have a very complex physiology. As I said, that they are social microbes. They do adventurous and social gliding motility. They make fruiting bodies. They secrete like a large number of secondary metabolites. Actually, they secrete more than actino and bacillus. They degrade a lot of biomolecules. The only problematic thing with them is they are very slow growing. They take like, their generation time is like almost 7 hours, which makes them, they don't grow very easily. So that's a very special negative point about these. But as a computational person, this is very fascinating because they have one of the largest genomes. So if you take, these are the all bacterial organisms, and these are the myxobacteria in the red. So they are exceptionally large, 9 to 16 MB majorly. You can see 1 or 2 or few here, but majorly are very large. Okay, and obviously because of that, they have a very complex signal transduction because they have to live such a complex life. Okay, now if you see they are natural pharmaceutical factories, and so this is the Myxococota, as you can see, and you will appreciate it from, these are all different phyla, and they stand actually very tall as compared to other organisms. And similarly, if you see their distribution per organism, these are actually one of the largest, this Pendulospiraceae. This is the recently identified genus which has the largest number of secondary metabolites according to anti-SMASH and so many other tools. The last feature which I will talk about today is their multicellularity behavior, and as several other organisms also show this kind of multicellularity, but their multicellularity is very complex with patterned multicellular structures like biofilms and swarms, and which require self-recognition. So they do self-recognition, spatial morphogenesis, cell differentiation, division of labor. All of these features are present within these organisms. So a very interesting model organism to work on in terms of evolution. Another thing which we have found very interesting in these organisms, which is again goes back into the evolutionary biology, is finding connecting link organisms. So now if you see, we have delta proteobacteria, non-myxococales, and if you see their genome size, which is usually 5 MB, they are anaerobes, extremely oligotrophic, and GC content is around 45% on average. And if you see other myxobacteria, they are 9 MB, aerobes, soil marine, and 70%. Now, if you see these 3 organisms, they have very low genome size as similar to delta. 2 of them are anaerobic in nature, similar to delta. 1 is aerobic in nature, similar to myxo, and even one of them have almost 45% GC content, similar to delta, and others have similar GC content as other myxo. So we are hypothesizing that these 3 can be very good connecting link organisms to understand the physiology and evolution of these myxobacteria. So we do a lot of, we have been working on it from last several years. Even I did my PhD with myxo and even postdoc with myxo. So myxo was the model organism. It's not PI. Okay. So we do a lot of work, and today I will be talking about a little bit into signal transduction part. Okay, so I'm sure signal transduction is not new to all of us. Every organism needs signal transduction mechanism. Bacteria especially have 3 types of signal transduction mechanism: 1 component, 2 component, and chemosensory system. In 1 component, ligand will directly bind to the protein, and the HTH, which is DNA binding domain, will target the genes and activate and repress the transcription. Now in 2 component, there are 2 proteins. Ligand will bind to the kinase part, and then kinase will transfer the phosphorylation to response regulator, and then it will target the activation or repression. In the chemosensory, it's an advanced system, advanced type of 2-component system. But so here there are the ligand will bind to chemoreceptor protein, which is a MCP protein, methyl chemoreceptor, and then it will transfer the signal to key A. Key A is a histidine kinase, and then it will transfer it to key Y, which is response regulator, and then response regulator will regulate different type of functions. So it can be motility, which we usually know that chemotaxis regulates motility, but from last 20 years, it's very well known that chemotaxis can also regulate several alternative cellular functions like developmental gene expression, biofilm, EPS, flagellar biosynthesis, so many. So what we have been doing, we are studying majorly these chemosensory pathways. Okay, this is like the broad goal of the lab. We call it like sometimes you must have heard of these knock-knock jokes. So we try to go to each and every bacteria, try to ask that what they are sensing, and they respond by these computational methods. Okay. So now if you see these chemosensory systems or 2-component system, you will find that Mixo stands very tall as compared to other organisms, and frankly, it's not directly proportional to the genome size, if you will ask, because we have found several myxobacteria which have very high genome size, but they have less number of chemosensory system, so it's not directly proportional to genome size. So here it has been reported in 2014 or before that, around that they have 8 chemosensory system, which is exceptionally large. And if you see, like the beauty with microbes is that most of these pathways are usually present in clusters, and that's why I like working with Mixo or bacteria, because you don't have to explore like the whole genome and randomly present genes majorly. So this is a chemosensory pathway, and which are all present together, all of these components which I discussed in here. Okay. Now, key A protein is a histidine kinase specific for chemosensory system, and we have used key A, and it has been reported also as a very good phylogenetic marker for chemosensory system. So what we did, we try to go into like almost so there are around 30 cultured myxobacterial genomes available, and obviously, there are so many MAGs, there are so many draft genomes, but 30 is the right number for complete genome. So we thought that, okay, let's just move into complete genomes and try to see how diverse these chemosensory systems can be. So we took key A protein as a marker. Again, means I don't want you to see everything here. Just see the colors, and each key A cluster is one type of system. So if you see, here we have xanthus, here we have stigmatella. Again, here we will have xanthus and stigmatella. Similar pattern will be all across. So it's a very clearly differentiated monophyletic clades for each system. And based on their synteny, based on their neighborhood, based on their architecture, along with phylogenetic clading, we categorized all these chemosensory systems into almost 14 type of chemosensory systems. So earlier there were 8 known, but we identified 6 novel extra chromosomal, extra chemosensory systems in these organisms. So now if you 8 plus 6, now there are 14. Okay. What we also did, so now these are all 14. They have each has different type of architecture, different type of arrangement, and they sense and they do different type of functions. As of now, or as of like few years back, the function of only 2 of these chemosensory systems were known. So first is key 2, which is a diff system. It is involved in fruiting body formation, and FRZ, it's in type 4 pili based motility for reversal of motility. Okay, but based on our syntenic analysis, we also identified that this might be involved in riboflavin synthesis regulation, because this system is very conserved across all the Mixo, and these 4 or 5 proteins which are involved in this function, they are always present in a syntenic manner, which gives an impression that they are definitely involved in something like this. And a few of our experimental collaborators are working on confirming these functions. We also try to look into their evolutionary pattern, that like how they evolved, and it was very fascinating that if you see most of them, they are present as one clade, which means that they are more of functional diversification or neo-functionalization of same type of system. And if you see the closest homologs, they are coming from, let's say here, cyanobacteria, or maybe for ECSS, they are coming from like actino or beta proteobacteria. But delta proteobacteria is nowhere, so that is for sure. The delta proteobacteria is nowhere here, which means that they are not present in delta, and they are directly coming from several other groups of organisms to this organism. And these group of myxobacteria are very well known for that they procure a lot of data, a lot of DNA from other neighboring bacteria. Okay. So this is great. We identified that all these myxobacterial CSS are located all around the genome. There is no conserved manner of their occurrence in different species. So we classified all of them per organism, and then we started looking into each category one by one. So what we found in 2018 so this particular ECS, CC5, it is only present in one organism. It's not present anywhere. So we looked into it, and we found that this particular protein is involved in this particular chemosensory system, and it has this particular PAS domain. So PAS domain is known to sense oxygen and this potential redox potential and light. So that was very interesting. We looked into the whole data, and we found that there is only one protein in all these 34 myxobacteria which has PAS domain with MCP signal. So there are many MCP signal proteins, as you can see, but this one is only present once. So we looked further into their architecture, and again, as you can see, they share. So this is the Corellococcus chemosensory system architecture, and we found that in actino or cyno, they have exactly same type of architecture in a very conserved manner present in them. Again, suggesting that they evolved from those organisms, and now they have more of this energy taxis cluster here. Okay. So till now everything was fine, means we were moving perfectly in our directions, nothing to flagella or motility. So now recently what Utkarsha found that these two organisms are also very far away from each other. This one belongs to Cystobacterini, and this one belongs to Sorangini. So if you see, this is Enteromyxobacter, which is here, and Labilithrix is here, which I was showing earlier. So we found that this system is present in these two. Now something fascinating came into. So this is, remember, this is Enteromyxobacter. It's one of the connecting link organism. Connecting link means which we discussed that they are kind of good indicator of evolution. What we found that these two systems are present simultaneously, and just upstream and downstream of these two systems, we have flagellar gene clusters. And flagellar gene clusters are again organized in a good quantity here, as you can see. I'm sure this audience doesn't need an introduction to flagella, but again, flagella is like encoded by 30-50 proteins, and even if one or if the core protein is missing, flagella will not be synthesized. So that's how it works. And if there is a chemoattractant, it will move forward. If it's a chemorepellent, it will do the reverse tumbling using this KY and FLIM interaction. So what we did, because that's what we do, we try to find a phylogenetic marker and try to generate this phylogeny using Ka protein. Okay, so we identified all these Ka proteins from all the cultured microbes only. So remember, like till now, we are only working with the genomes and not anything else. And what we found that these organisms, so there is this specific cluster F7, which is only present in these organisms, which are actually the Enteromyxobacters. So all of these organisms are Enteromyxobacter. This is the same cluster which we discussed like 2 slides back. And if you see, F7 is a very monophyletic clade here. Okay, if you can see here, it's a single clade which has Andromyxobacter and Labellithrix. Labellithrix is coming from very far away. Okay, so which is very interesting, and F7 is involved, is known to be involved. Igor Zulin's lab Igor Zulin's lab has done a lot of work on this field, and they have characterized all these type of flagellar systems very clearly. F7 is involved in flagellar motility. So this is sure that all these clusters which are together, they are involved in flagellar synthesis or flagellar regulation. Now, we also looked into all these orthologs and chemosensory gene orthologs, and we found that only in this group and Labellithrix we have this pattern. Otherwise, these genes are absent. Okay, so this is great. Now, this is surprising, and this will be, this is kind of a factor that Myxobacteria don't have flagella. Okay, Myxobacteria don't have flagella. They move by pili. They move by focal adhesion. So that is very interesting because they don't have flagella. Now, what's happening with this connecting link organism, Andromyxobacter? That was a very specific question, and we thought of it. Let's do what we can do. So what happened? Means just by surprise, or how the destiny takes a turn. We found this paper, which was just in 2023 when Utkarsha was doing all this analysis, and she found that we have some myxobacterial mags which has photosynthesis ability. Okay, so now if you know, means photosynthesis is like majorly limited to cyanobacteria. Okay, so now these myxobacterial mags, they also have the photosynthesis capability, which makes them very strange. They specifically mentioned that this potential of photosynthesis is, or the genes of these photosynthesis are only present in mags, not in genomes. Okay, so not in cultured organisms, but only in mags. Okay, so obviously for that there cannot be no experimental validation because they are not in genomes. Okay, so we thought that okay, if we are finding such kind of pattern in these mags, why not let's take these mags and try to find out about this flagellar system. So we did the same thing. We took all those mags. And now Utkarsha is extending it to all available mags, which are like almost 5,000 mags for Myxobacteria. But at least for this analysis, she took only these 18 mags which they have studied. And what she found, that again, there is a very well demarcated monophyletic clade for this F7 system. As you can see, this is F7 and this is F8, which was the nearby one. And these red ones are the mags. Okay, which is very fascinating that these mags have the same type of Ki protein, and they also have the F7 flagellar systems. So now we looked into each of those mags, and and we found that many of these organisms, they have, so if you see the star ones, they are the mags, and the without star, those are the genomes. Okay, or you can say the green ones are the genome and red ones are the mags. Okay, and we found several of these flagellar genes. Remember, like mags are draft genomes, so we cannot trace like the whole patterns like in genome, but we were still able to find several chemosensory genes in between and flagellar genes on both ends. So which is very interesting that most of these mags, 18 mags, like almost 84% of those mags, almost like 15, 16, they have CSS and flagellar gene ortholog in the same content. Okay, so means again this is a very bulky figure, but just want you to focus on a few things. So the highlighted ones are the mags, the non-highlighted ones are the genome, and the non-box ones like here or here, these are the other organisms which don't have flagellar genes or majority of the flagellar genes. So now if you see, most of these organisms which are in the box, they have most of the flagellar genes, obviously not all of them. These are all the core genes here for flagellar synthesis, and they have some of the core genes absent, which makes sense because Myxobacteria nowadays don't have flagella, so they have lost some of the core genes. Now we also looked into their their chemosensory system identification, like which one they have. And if you see the orange color, if you can see the orange color depicts the F7 system, and this F7 system is only present in those organisms which have the flagellar genes. So this is chemosensory system, this is flagellar system. Okay, so flagellar, so we can easily correlate that there is a very good correlation between flagellar system and the chemosensory system presence in both of them. We went into one more step further. So you remember the MCP protein, which I discussed in the first few slides. MCP protein is the protein which senses the ligand, and based on that, it will do the move. It will allow the bacteria to do the movement. So we found that this 36H is the protein which regulates flagellar motility, and this protein is exclusively present only in those organisms which have the flagellar system and the respective chemosensory system. So there is a perfect correlation between the presence of flagellar gene cluster with chemosensory system with the MCP protein, which will sense and allow the chemosensory system to regulate the flagellar system. So based on this, this is our, like putative, very vague model. As of now, that, there were delta proteobacteria which evolved into the current world cultured myxobacteria, along with these uncultured myxobacteria which are present in like some old world territory. And these organisms, the connecting links and these uncultured myxobacteria, they have the flagella and the chemosensory system, which is responsible for their motility. But due to so many evolutionary pressure and changes, and maybe their residence or the habitat change, the present world cultured myxobacteria, they lose the flagella and they also lose the associated chemosensory system altogether. Okay, so this work is still in progress. Utkarsha is trying to explore all the MAGs and all together and try to come up with the, like what we will find. But yeah, so that is the current hypothesis or the conclusions. And with that, I would just like to thank all the people who have been working with us. And means we are always hiring at IIT Hyderabad. So in case anyone is looking for positions, feel free to approach. And special thanks to DST and IIT Hyderabad for funding. And I would just leave you with this, old paper and old quote from Carl Woese, that the time has come to replace the pure reductionist eyes-down molecular perspective with a new and genuinely holistic eyes-up views of the living world, one whose primary focus is on evolution, emergence, and biology in its complexity. And yes, means this conference or this symposium is definitely following Carl Woese. So yeah, all credit to him. Thank you.
Thank you, Gaurav, for finishing in time. So any questions in the audience?
Thank you very much, Gaurav. I was wondering, myxobacteria, of course, also harnessed for a lot of natural products.
Yes.
And I was wondering, if you mention that they've lost their ability for social interactions, the natural products would be suggestive of the fact that they are indeed interacting a lot.
No, but they are doing a lot of social interactions. They didn't lose it. They have the social interaction ability. And that's where these metabolites come into picture, because they usually secrete them, and other nearby organisms, they outcompete them for their own food and all.
Right, okay. Yeah, so there's a large diversity, I guess, of social interactions that they are actually doing. Yeah, thank you.
Means they also have the kin selection and so many other features which makes them, where they utilize all these antibiotics and all.
Thanks, Gaurav. Interesting talk. So what I wanted to understand was, do you see a transition from flagellar motility to gliding motility, or was gliding motility always there, the pili?
Yeah, that's actually a very good question. So we looked into it. So as of now, it looks like even these old world myxo or the MAGs, they also have pili genes. The current world also have pili genes. But for flagella, it's definitely very, black and white, that the genomes don't have it, like the larger ones, which I showed, except the connecting links, they don't have it. They don't have that chemosensory, like the whole chemosensory system got lost. Or maybe you can say, slowly it has lost in them. In connecting links, it is still present. Maybe they will lose it in next million years or what.
So do you see, for example, any transitions in terms of the metabolic capabilities of these mixo? Because maybe flagellar motility was required for certain specific substrates and...
That's good point. So Enteromyxobacter has this faculty, Enteromyxobacters are facultative anaerobe. Okay, so they can stay in aerobic environment, they can stay in anaerobic environment. And the idea which I have is that even these mags, they are staying in kind of, like a deep ocean or some other places. So maybe in that kind of environment, it was required. Maybe now it's not required because now they are living in soil and they can easily glide. So yeah, that is the only explanation which we have as of now.
Yeah, we have one online question. So it's like, if we have only a subset of genes is present, what is the effect on function? And how different can the function become?
Yeah, so subset of genes is just an indicator that that function would be there. So now it depends on from which perspective we are coming. Either it is gaining that function or it is losing that function. Okay, so if you have just 5 genes out of 20, it's possible that there were 20 and now they have 5, or it is possible that they have 5, maybe they will, or they were not able to procure like all the 20. So yeah, it's a more of a reversible kind of thing. So it will have, if we have just subset of genes in a complete genome, okay, so that clearly means that that function cannot happen because that is subset. We need the full set to have the function. But if the subset of genes is present in a draft genome, so draft genome is incomplete, so which means that there can be other subset in the rest of the draft, making it that that function can be there. Yeah, I hope that explains whoever asked that.
So I have one question. So you have mentioned that it has been evolved from the cyanobacteria. So what is the natural habitat of this myxobacteria?
So all the cultured ones, they usually remain in soil. They are soil bacteria. A few of them are marine. Few of them are also, coming from like different type of, like cryo kind of places like Iceland and all. But majorly they are soil microbes.
Thank you. Thank you, Gaurav.
Good morning, everyone. So, my name is Suresh Sudasan. I'm working from the Novo Nordisk Foundation Center for Biosustainability. But today I have the pleasure to introduce the next speaker, Dr. Anshu Bharadwaj. She is a principal scientist at the CSIR Institute of Microbial Biotechnology in Chandigarh, India, specializing in bioinformatics, systems biology, and infectious disease research. She obtained a PhD from the Center for Cellular and Molecular Biology at Hyderabad. And Dr. Bharadwaj has developed several predictive methods, databases, and ontology-based barcoding techniques for genome variation data, enhancing the understanding of genotype to phenotype correlations. Her accolades include being recognized as a young innovator in India by the United Nations Development Program and selection for the International Visitor Leadership Program by the US States Department. So today, she is here to talk about how data-driven approaches can be leveraged to understand and address complex diseases. So with that said, we are looking forward to your talk. Thanks.
Thank you so much for that wonderful introduction, and I'm really glad to be here. Thanks to both the Kartiks for introducing me to this fantastic community of researchers. I'm a new person to microbiome, so let's see what we can discuss today. Am I audible to everyone? All right, great. So since I am a new member to the community, what I have done today is to introduce you to what kind of work we do in the lab. So I'll be going very fast, so pay attention. It's like 30 minutes of flash talk. I'll be covering on a lot of stories, so let's see how much we can discuss today. So my interest and my lab's group interest is also to explore genotype-phenotype correlations and to understand antimicrobial resistance as well as mitochondrial diseases. We also are heavily involved in doing computational drug discovery from different perspectives. So I'll keep picking up a few examples from the work we have done in the past, some ongoing work and something that we intend to do in future. So we use a variety of approaches in our lab to address AMR and mitochondrial diseases, including next generation sequencing, systems biology, to use network medicine. We also use a lot of ensemble-based machine learning methods to address a lot of issues that we face in understanding AMR and also trying to find solutions to it. Now, I don't need to introduce antimicrobial resistance to anybody in the audience. We all understand that it's a silent pandemic. But what I want to convey is that the mechanisms the bacteria adopt in response to antibiotics are very, very complex. They are not very simple, and that is where the complexity of genotype-phenotype correlation arrives. And this is where we try and understand, can we find the Achilles heel or the weaknesses in the system which can be harnessed for discovery of new drugs. So when we started working on AMR a few years ago, our model system was tuberculosis and then MTB. And what we intend to do was, we were trying to see if we can use a data-driven, unbiased approach to understand or find new drug targets in MTB through a more systems-level approach, which is more knowledge-driven. Okay? So we did a very big crowdsourcing activity to manually curate the protein-protein functional network and the metabolic network of MTB. I'll not discuss the protein-protein interaction network today, but I would like to focus on the metabolic network. Now, I'm thankful to the speakers yesterday because it made my job easy. I need not explain what flux balance analysis is. But we are used to seeing metabolic networks like this. So one thing we did was to simplify this representation, and we started representing the metabolic models as a vertical of exchange reactions, followed by all the metabolites listed, followed by all the genes and the reactions which they actually carry out. When we mapped all the curated reactions that MTB is capable of doing, this is how the network looks like. This is a spindle map. So these are all the interactions that MTB is capable of. How we used this network was, we actually started doing these in silico gene deletions to find which of them actually does not lead to creation of a biomass, thus telling us that these genes are important from the metabolic point of view. Now, this is the entire metabolic capacity of MTB, right, with 890 genes. But when you grow this in, like when you do flux balance analysis and grow it in the wild type condition, you see that only half of it is required by MTB to normally and happily grow. It is not using all of the genes it actually is capable of using. So the flux through the rest of the reactions were 0. So in this scenario where we saw happily growing mycobacteria, we did a knockout of the isoniazid drug target, which is INHA, and what it tells us that it actually leads to a tremendous information loss, which probably explains why isoniazid works as such a wonderful drug for TB. What we then saw in this particular condition is that there were at least 60 genes that started showing flux in this knockout background, and they were not showing any flux in the wild type environment. Now, these are the alternate routes of metabolic rerouting that mycobacteria is adopting to basically render a phenotype which is called as persistence. It's a very difficult phenomenon to understand, and that is where the entire biology of mycobacteria is currently going. So in order to confirm what we observed, we look at isoniazid exposed expression data sets, and we found that 42 of the 60 genes that we are now finding to have a flux through them also are overexpressed in isoniazid exposed mycobacteria, rendering confidence that what we are observing here as alternate mechanisms of metabolic rerouting are actually happening in MTB, at least in vitro cultures. And we then identified that the fatty acid metabolism seems to be the backbone of mycobacterial backup mechanisms that are rendering it persistent to a lot of antibiotics that we are developing for it. So this is pretty much, so as I told you, I'll be covering a lot of stories. So this is all what I'm going to tell you about this story. Now, subsequently, we started applying the same set of methods to a lot of other organisms, including M. abscessus, which is an NTM. We discussed why NTMs are important to study. They're an emerging pathogen space. We are also applying the same thing to Acinetobacter baumannii and a lot of other pathogens. What difficulty we face is that in most of these cases, the genome annotation is so poor that we cannot develop a very comprehensive metabolic reconstruction or to do anything with these kind of annotations that are available. So what we did, we actually utilized the outcomes of the recent Nobel Prize winning discovery. We saw that, okay, can we use AlphaFold to our rescue, which allow us to do a more refined annotation of these genomes that we can then use for systems level modeling or other analysis. And we took M. abscessus as a case study, and you can see that out of around 4,900 proteins, 1,000 of them actually don't have any function. Even the others which have function are not very finely mapped. So since structural based alignments are computationally very expensive, we actually took the help of fold-seq, which is a very recent method. So what you are doing is AlphaFold utilizes protein sequence, converts it into a structure. Fold-seq utilizes structure and looks at the tertiary residue-residue interactions and represent those structures as 3D states or alphabets. So you are going from sequence to structure and back to a sequence of 3D alphabets, rendering this entire process extremely fast and can be scaled up to a genome scale level. So we did this analysis, and if you see there, this is the 3D, this is how 3D alphabet looks like, and this is basically each amino acid is mapped to their own 3D space, and this is the alphabet representation. Now you can actually use these alphabets to do language models, and we have used Prostify in this case wherever fold-seq failed to give any structural alignment. So this is where you can actually utilize the foldy 3D alphabets to also do large language model based analysis. What did we get out of it? So what I'll give you is a very simple example. So we took a protein and MAB, which is considered to be an essential protein from the transposon-seq experiment. But if you go to UniProt or any other literature, you find that this has very poor annotation score. There are no gene ontology terms associated with it. So you know from a transposon-seq data that it is an essential protein for MAP, but you don't know what it is doing there. So with our method, we did a lot of sequence-based annotation, but didn't find an exact function for it. But when we did a structural-based annotation using fold-seq and cath, what we saw, it actually mapped to the MTB had AB complex. Now just briefly, what does this complex do? This complex is a dehydratase, which actually is very critical for mycolic acid formation in mycobacteria, which is absolutely essential for mycobacteria to survive, explaining why it was coming essential in the transposon-seq data. When we overlapped the HAD-A-B complex from MTB and MAP that we predicted, we saw a decent overlap, but there were also differences. One difference which is actually notable is the 1661 position, because it has been seen that when 1661 changes to serine, it actually renders drug resistance in TB patients. So this is a mutation that is directly associated to resistance in MTB. In MAB, there is no 1661, instead there is an alanine. So that you actually know that the flavonoid inhibitors are not going to work in MAPSIS. And this is how going from sequence to structure, having a very finely defined function to it is allowing us to also prioritize targets and take it forward and basically enhancing our understanding of these microbes. So we developed an entire pipeline to do this, and we are applying it to all the WHO priority pathogens right now, and its work is under review. As I told you, we also do computational drug discovery. So I'll not describe this today. This is how we actually do a more structural based modeling kind of approach. We are not discussing this, but I would like to discuss this method that we have developed. So this is basically drug repurposing. The rate at which we need antibiotics and the rate at which we are discovering antibiotics is like completely out of phase, and we really need to speed up the discovery. So we thought, let's try doing drug repurposing. For the audience who don't understand this, if there is one drug which has been approved for a particular disease because you know the target, and if some criteria tells you that the targets are similar or the drugs are similar, then drug 1 can be repurposed for disease 2. Likewise, drug 2 can be repurposed for disease 1. So you are repurposing the indication of the disease condition. Why do we think it was possible? If you look at the drug bank data, which is the repository of the drugs that are being used in human, you find that it's a very small percentage of drug that have a single drug gene interaction. One drug interacting with one gene. Most of it, like more than 90% of it, is one drug, multiple targets, or one target, multiple drugs. So its promiscuity is built into the way we discover drugs, which now you can harness to see how can you look at this network and identify how do you identify new targets for the known drugs. So we used a network-based inference approach. I'll not describe this. If you are interested, we can talk about it. Focus on this part of the slide. So these are the known drug target interactions. The logic of doing NBI is to identify the new ones. So can you add new edges to this network? And this is basically an NBI resource propagation technique, and that is what we applied in this context to identify those new interactions. Once the new interactions are identified, then we applied several target prioritization methods, several drug prioritization methods to basically recommend drug-like candidates for a lot of priority pathogens. This is what we have currently finished. What we notice is that the drugs that we are finding, as in the new drug targets, and the drugs that we are finding belong to a large number of priority pathogens. Some of them are broad spectrum, some of them are narrow spectrum, and with this we have been able to basically, shortlist some of the drug candidates for the priority pathogens recommended by WHO. Now, if you remember, in the NBI, we have not used the structure of the compound, we have not used the structure of the protein. There is no structural data, we are only working on their interactions. But if we then look at these targets, and then you look at the known drug and the predicted drug, they actually share structural similarity. And so this actually lends credence to the fact that the method probably has worked. And for some of them it's rush show, for some of them it doesn't, and it's actually a good thing because you're looking at new scaffolds and not the ones that you already have. So that's something that we did in this work. Now, very quickly, I'll finish this part of the work where while we were doing small molecule drug discovery in a target-based or in a drug repurposing-based strategy, we also realized that the biofilms are the most common way in which bacteria actually exist in nature. They are not existing as planktonic versions. So they actually know their natural lifestyle is like biofilm architecture and not a planktonic one. And these are basically microcolonies, and you have one or more than one bacteria inside each biofilm. Depending on where the pathogen is, either they are metabolically active or they are slow growing, and that is why you need different combination of antibiotics to treat them. To the best of my knowledge, there are no currently available antibiotics to deal with biofilms. So this is something which is still an open area of research. They're very adaptive in nature, and they have the resistance mechanisms that you understand in planktonic cultures is very different from what operates in a biofilm environment. So then we were like, okay, how do we design something that can be used in this context? So we actually took help from nature. So what we as hosts do is we make actually the defense peptides with our cathelicidins, and they can disrupt biofilm structures. So then we realized, instead of a small molecule based approach, why don't we actually take help in terms of making or designing peptides that can inhibit and or basically function as anti-biofilm at different stages of biofilm life cycle. Now in this case, we applied machine learning, which is again a different method, and it's applied in a very different context here. So the positive data set in an ML, in this case, is biofilm disrupting peptides. We know that they disrupt biofilms. And the contrasting positive set are biofilm maintaining peptides. These are not the peptides which are inactive in a biofilm assay. These are the ones which are actually responsible in quorum sensing in maintaining and creating the biofilm. So it's like an absolutely contrasting set for ML to learn from. And then we saw whether is this working or not. And then we applied supervised learning. We developed several models, but just to tell you, all these models are based on only the sequence of the peptides, not the structure. Sequence enough was good enough to tell us which sequence portions are good for basically making an antibiofilm prediction. Now just to tell you, these are the model performance on independent data sets. But to tell you in more biological sense what might be happening, we all know the EPS, which is the extrapolymeric substance. It is negatively charged because there is DNA in that, right? So one would assume that antibiotics like tobramycin, which are positively charged, can be sequestered deep into the biofilm. But actually that does not happen, because via some ionic interaction with unknown components, tobramycin gets sequestered, and positively charged antibiotics don't work in a biofilm architecture. Negatively charged aminoglycosides obviously will not work because of the charge-charge incompatibility. However, neutral antibiotics like ciprofloxacin actually render deep into the biofilm, and that is what I think our model studied, because the peptides that it predicted, which are of antibiofilm in nature, are hydrophilic. So this is why I think why the model worked very well, because we could prioritize hydrophilic nature of these peptides, which can actually go deep into the biofilm architecture. We then applied it on FDA-approved peptide drugs and some of the AMPs in clinical trials. And the original indication tells you that these are the sites of biofilm formation. So this is again a drug repurposing, where we are saying that these peptides can now actually be developed also for their antibiofilm nature, not just as antimicrobial peptides. You can test this application. A lot of people in the world have used it, and it really works in an experimental setup. So I would like to have your comment on if you use it at some point, tell us how to improve it further. All right. So coming back to what we do, so what I have done is I have picked up a few examples from here and we discussed that. I would like to now very briefly discuss the below portion where we work on mitochondrial diseases. So now why we got excited about mitochondria is because of the extreme genotype-phenotype complexity. Now mitochondria is a cellular organelle that carries its own DNA and not just one. So each cell has several mitochondria and each mitochondria has several mitochondrial DNA. And when the mitochondria has to function, a portion of the genes are coming from mitochondria, but a very large portion of those genes are coming from the nucleus. Because of heteroplasmy, because of a lot of other things, establishing genotype-phenotype correlation in mitochondrial disorders is extremely hard. Our group has been developing a lot of methods over past years to resolve this, and I'll not discuss this today. These are all published work. But what we are doing it from a more translational point of view is, we are part of the NIH Clinical Domains Working Group, and we actually help the clinicians basically make guidelines for the world to say how the variants in the mitochondrial DNA can be used in a more clinical context. Now this is important because it's only in 2016 that the Mitochondrial Research Society said that you should do sequencing as the first line of diagnosis for mitochondria. Before that, sequencing was the last step if at all you want to do it. It was not the part of the clinical workflow, which it is now. Because of next generation sequencing methods, you can understand heteroplasmy. And that is when the decision was taken to push it as the first line of diagnosis, and that is what we do with the NIH and the ClinGen teams. But here, today, I want to keep the focus on antimicrobial resistance or not mitochondrial diseases. So we are exploring mitochondria from an infection predisposition point of view. Because we all understand that mitochondria is the organelle which is responsible for cell fate decisions, energy status, immune responses. So it is a key battleground for host pathogen interactions. Pathogens are very smart. They hijack the host mitochondria to establish infections. And that is what we try and probe in this work. And what mechanism we are focusing on are the mitochondrial quality check pathways. Now the mitochondrial biogenesis and mitochondrial degradation, like on an average of 1 month in the brain, the mitochondria recycles, right? Now this recycling is something that the pathogens have understood, and then they try to control this cycle so that they can render the environment in the host favorable for them to replicate. Right? And this is what we intend to probe. So we actually were trying to probe mitophagy, which is a mitochondrial degradation pathway, and trying to understand how pathogens are actually exploiting it. So what is the hypothesis here? So the hypothesis that we built is that if a pathogen has to establish infection, then the protein in the virus or the bacteria should be a low central protein because it is free to evolve, right? So it should be under positive or neutral selection. The candidate in the host which it is targeting should be under purifying selection, because that particular host protein should not be freely evolving. So it should be under constraint. So this should be high central proteins, this should be low central proteins, the host protein should be under purifying selection, but the pathogen protein should be under neutral selection. Now we wanted to test this hypothesis. And what we did, we curated mitochondrial associated host pathogen interactions across all viral and bacterial data that is available today, and we have 37,000 such interactions that are curated and we know that they exist. Then we apply centrality measures on the human binary interactome and identified that 26 proteins which are known to play a very important role in mitophagy are in the top 1% central proteins in the human binary interactome. So you know that this is a central protein for the host. I'm not giving you more examples here, but just one story. So this is a case study. So our hypothesis was we were trying to understand a host protein which is under purifying selection and is very central. LRRK2 is one such example, which is a leucine-rich repeat kinase. It is one of the hallmarks of how Parkinson's disease actually manifests. And why I'm talking of Parkinson's disease and infection, it will become clear in another 30 seconds. Now LRRK2 is a protein that interacts with a meroprotein which happens to be on the mitochondrial membrane. Now mitochondria inside the cell are mobile. They keep moving inside the cell. Whenever a mitochondria dysfunctions, right, and it starts to lose its membrane permeability and there is issues, then it is dislodged from that movement and it is marked for lysosomal degradation, right? In Parkinson's disease, exactly this does not happen, and that is why mitochondria keeps generating ROS, putting the system under pressure and hence leading to localized necrosis or apoptosis, and that is what leads to tissue degeneration. That is the way how Parkinson's happen. Now, LRRK2 in a wild type scenario, whenever they see that a mitochondria is going dysfunctional, they come and degrade this meroprotein. The mero tethers the mitochondria to the kinesin. When this degradation happens, mitochondria is out of that cytoskeletal machinery and is marked for degradation. In a Parkinson's disease phenotype, when the LRRK2 is mutated, this mutated LRRK2 cannot degrade mero. The dysfunctional mitochondria keep circulating in the cell, releasing ROS, and that is how the disease manifests. Now, it happens that E. coli and dengue virus also interact with LRRK2 in a similar manner. And a longitudinal study clearly shows that patients with dengue fever exhibit a 2.59-fold increased risk of developing Parkinson's disease. So, two very different phenotypes, one is a neurodegenerative disorder, the other is an infection, actually tends to have a common molecular basis, and this is exactly why we thought that we'll explore these comorbid conditions from an infection perspective, where we can utilize this data for host-directed therapies and prioritize them in many different ways. So, we are actually focusing on bacterial priority pathogens again here, and we are also trying to understand these HPIs in a very population-specific manner. The complexity of this whole scenario increases because now we are talking of human pangenomes. The human reference is not enough, and now we know why precision medicine is going to be even more complicated. So, this is basically, I kept a reference for the students. We wrote an editorial which actually captures the past 22 years of human genome to the human pangenomes, and it will be nice if you can read it. Now, in the last few minutes, since it's a microbiome conference, I cannot conclude the talk without telling you what we do in microbiomes. So, what we are currently doing, there is an ongoing Phenomedia CSIR Health cohort knowledge base. It's a very large project going on, where we are sampling in a longitudinal manner 10,000 CSIR employees over a period of next 5 years. We are doing a lot of body scanning, blood parameters, a lot of omics, to understand how longitudinally we can actually make risk prediction for different disease conditions. Okay? One of the core activities in this exercise is to understand the gut microbiome in the human population. We have finished the first set of sampling in this project, and we have now whole genome sequences of 6,000 odd individuals microbiome data from the Indian population, and we are analyzing it. And we also are giving a report to all the individuals who actually got their sample sequence in terms of their diversity, the F/B ratios, and what kind of species do they have, so that they understand how their gut health looks like, and how we can then give them advice in terms of lifestyle changes and see after a year, after 2 years, after 3 years, how this actually changes the gut microbiome to a more healthy, diverse profile. So that's an ongoing project. Apart from that, we do a lot of science outreach and engagement in terms of doing classroom experiments. We make a lot of online videos. But something that we do a little differently is this. We also develop video games to generate awareness on a lot of topics of interest. So Armor is a video game on generating awareness on antimicrobial resistance. It's a multi-level game targeted for school kids, so that without feeling the burden of understanding this complex phenomenon, they'll play and learn and might influence behavior change in them. It is endorsed by WHO, and we were also one of the top 5 games in the world in the global competition of ECGBL in 2021. And Armor was an outcome of a lot of homework that we did to understand what's missing in the current AMR games, and that is also what we have published recently. And this is, it's a multi-level game, as I told you, so maybe you would like to go and check it out. What I'll do is I'll try and play a small teaser to the game, just to give you an idea of what it intends to convey. The sound is not coming across. I'll try and see if I can use this. So all right, that was the teaser to the video game. The video game doesn't look like that. So how does it look like? I would like you to go and give your feedback. Right now it has 8 levels. But when we were developing Armor, we also realized that we could develop something which is also a multiplayer game. So what I'm going to show you in next 2 slides is something that I haven't shown to anybody so far. So that's a new game that kind of a semi-launched here. It's a game on gut microbiome. And I thought what better than the microbiome community to show it to. So that's the game which we call as gut feeling. And that is how the interface looks like. It's a you can play it as a single player. You can play it as a multiplayer on the cloud. So your partner can be sitting anywhere in the world. And what is the game idea behind the game is to basically incentivize, again, school kids. It's easier for them to change habits, to opt for good food options, understand why it is important for the gut, and then utilize this knowledge in their lifestyle implementation in terms of not falling sick. The game is supposed to have 8 levels. Since it's an ongoing work, we have finished 4 game levels, and there are 2 mini games in between, which actually are important because that gives additional points for the user to win the next levels. So the gameplay is like a maze. So every time you open the game, even as the same player, you will see that the maze design has changed. So the maze design keeps on changing, and you can play it as a single player game or as a multiplayer game. You have to make good food choices, and so you need to find how do you traverse from here to the endpoint, but you also have to do making sure you avoid all the bad food options. Now, in order to make sure that what we are popularizing at good food choices, we have a lot of Indian food items there, because if you look at this report by WWF, they are saying that Indian food is the one which is very climate sustainable, and that is why we thought it's not a bad idea to also, popularize what choices do we have in India, what do we eat, and why it is so gut friendly and why it is also so environment friendly. So the game rules are very simple. So you are assigned an avatar based on what your lifestyle is currently. So whether you will have like a macho avatar or you're going to have a weak one or an obese one depends on what your lifestyle is. And then you have the catch that the shortest path in the maze has unhealthy food options. So if you're very keen on winning the game, you are actually not winning it because you are thrown with things which are not very good for the gut. So you need to navigate and find the best path where you actually are marked for food diversity and not just how quickly you finish the game. And if you have made unhealthy choices, there are provisions in the game where you can exercise and get away using the same path or change the route. So we are trying to say lifestyle, both in terms of food and exercise, are important. So that is what we have shown in the first 4 levels. The subsequent 4 levels are going to be the different axes that we keep talking about, the gut-brain axis and stuff like that. So that will be done by next month. So I'll not play this entire onboarding video, but maybe 5 seconds of it.
Do you think you can make the best food choices for a healthy gut? Then join us in the ultimate challenge.
So there's a full onboarding video, but in the interest of time, I'll not play this. All right, so I work with a lot of community which are not sitting in my lab all the time, and they cannot fit on the slide also. So, but I would like to thank all of them because it's really interesting to work, and I've been working extensively with school kids and not just for science outreach. These are kids who have done very serious scientific projects in the lab, almost equivalent to what my PhD students do. So this is why I keep showing, this is a slide for the students out there. There is no age to do science. There is no degree to do science. These kids can really handle very, very complex projects even when they were in school. So, the landscape of doing science is changing. Is just to convey the message. Pooja has just finished her PhD from York University in cryo-EM. Ashma is now at Caltech doing her PhD in trying to understand the origin of life. And they have published papers with me when they were finishing school, like 10th standard, 11th standard, 12th standard. So they are very, very young when they really did some outstanding work. And with that, thank you very much again, both the Kartiks. It's a wonderful opportunity to come here and talk to so many people. I'm just enjoying myself, that much I can tell you. So thank you so much.
Thank you, Dr. Bharadwaj. It's an exciting talk. I have now something to take home for my little daughter. Yeah, good. So, any questions from the audience?
Hello, madam. Very interesting talk and nice games. I just wanted to ask you one basic question, like when we do drug repurposing, so normally when you do from novel drugs, so you go from heat to lead and then do it in animal testing, and then you have a phase 1, 2, 3 clinical trials. So what are the advantages of drug repurposing? So we need to go through all these steps again, or can we skip some of these steps?
Yeah, so the advantage of drug repurposing is that since you're working with drugs that have seen, you have done animal testing, you also have done human testing, so the pharmacokinetic profiles of these drugs is worked out. So you know at what dosage you can play with it. Like I'll tell you a very simple limitation. When you take anti-diabetic or other drugs, anti-diabetic still okay, like you still take 500 milligram of metformin. But if you take an anti-BP or other drugs, they are very small in how much dosage you can give, like 50 mg. But normally when you talk of antibiotics, they're like 250 mg, 500 mg, 1 gram, right? So, but since you have worked out the pharmacokinetics of the drug, a lot of early stage drug discovery, a lot of hit to lead optimization, those and GLP tox, which is one of the most expensive process in drug discovery is GLP tox. It's very expensive. That you can bypass, and then you can actually do dose optimization in a preclinical model and take it forward. So that is why you actually save, if not less, at least 7 to 8 years of work plus the cost. Because the drug discovery attrition rate is high because of multiple reasons. Cost is a major limiting factor when you are talking of antibiotics.
Hello. Hi. Very, very interesting work. I had a question about the study on the 10,000 individuals in your organization with the health of their gut microbiomes that you're giving a report on. I'm wondering how you're defining the normal gut microbiome of the Indian population.
Good question. So that is exactly what the whole study is about. See, we have been following all the blood parameters or like the scanning parameters that is coming from the Caucasian population. We don't even know the normal in India. So we don't know how do we define normal in India in terms of cholesterol or in terms of any other blood parameter. So what we are trying to do here is to sample decent, across the country and then try to understand what normal means. We don't yet know that. For all practical purposes, even the F/B ratio, we have assumed it to be, 1:1. But if you look at other studies, they show it's 3.7:1. So there is no consensus on what normal is, to be honest. We are just trying, since we have the questionnaire from the participants, we know if they are struggling with any disease condition. We try and do unsupervised MLAI techniques to see what do we see from this data. So we are not making any assumptions on what normal is in this case.
Thank you.
Hello, ma'am. Your talk was nice, and also the gut microbiome part is also very good. I have one, question like this. Can we model the gut microbiome of the individuals and their lifestyle, their feeding habit and all, and do some machine learning type things and also predict some personalized feeding habitat for them for maintaining a healthy lifestyle?
Yeah, it's a very relevant question, and I think all the speakers you heard yesterday who are talking of how communities interact in the microbiome. See, what we are doing in a microbiome data analysis in this case is we are just doing a diversity mapping, but that is not how the gut is maintained. Like, it's a very dynamic environment. So I think methods that this group is, actually this community is building, along with what we are building, can actually be pooled together to say how do we integrate lifestyle choices to the dynamic nature of the gut. And that is why this longitudinal data is being generated, because a single snapshot will not help us understand what's happening. But I think the timelines that we are referring to here, which is like 18 months of sampling, is not ideal, because gut is dynamic. We should actually have more depth longitudinal data over a period of time. And then when each one of them developed by Dr. Narendra or Karthik and other members here, if we can now pool those techniques into this data, it will give us indications of what are the keystone species that you need to maintain, because it's just too much of data out there. And then how do we then fine tune that, because we know which metabolite can come from which food item. Yes. And then which individual might need what kind of food intervention to stabilize the gut.
Yes, ma'am. Thank you.
Thanks, Dr. Bharadwaj. All right. Before you go on for the tea break, there's one exciting speaker here. So Paul Williams, Professor Paul Williams here, is a professor of systems ecology at the University of Luxembourg. He earned his PhD from the School of Environmental Sciences at the University of Anglia in Norwich, United Kingdom. Dr. Wilms' research focuses on the study of mixed microbial communities such as the human gut microbiome and their interactions with the environment. During the COVID pandemic, Dr. Wilms led the national response by the Luxembourg scientific community, resulting in Luxembourg having the lowest excess mortality due to COVID in the EU plus UK. He has also authored over 150 peer-reviewed publications and is a highly cited researcher. Dr. Wilms has received numerous awards and prizes for his scientific contributions, including a European Research Council Consolidated Grant in 2019. So today he's here to enlighten us on the talk from molecules to impact systems ecology of the human microbiome. Dr. Wilms, the floor is yours.
Thank you very much for that kind introduction. And also from my side, thanks a lot to the organizers, in particular Kartik and Kartik, for this wonderful meeting. So yes, I will talk a bit about molecules and what impact they might have on the host side, and in particular, how they might be triggering disease processes. So we still think that these are very exciting times by which we are able to perform systematic measurements of microbial communities, so integrating metagenomic, metatranscriptomic, metaproteomic, and metabolomic data. And I think what is really crucial here is that we couple these analysis tools to clever study designs in relation to particular longitudinal and spatial sampling. And if we do this in a proper systematic manner, we should get to the point of being able to reconstruct the constituent networks of individual populations, such as illustrated here by metabolic networks, and importantly, understand through the analysis how these different populations then interact, and in particular through those ecological interactions, how that gives rise to emergent properties that themselves may in fact impact the host. So we might see molecules that in isolate culture you would never see, of course, that would be the result of these interactions, and that may indeed impact host physiology, most notably by triggering disease processes in susceptible human cell populations, such as in neuronal cells or indeed in immune cells. And so the molecules that are involved, I've just touched upon small molecules, but these come, of course, in different shapes and forms. They range from nucleic acid moieties all the way then also to small molecules. And we've recently coined this cocktail of biomolecules as the expo-biome. We can have a long-term discussion about terms and terminologies, but at least we've given a name to it. And it's, in our mind, important to look at this whole complexity, because the complex cocktail of biomolecules would be what would confer pathogenic properties to microbiomes. But whether that's the case, of course, or not, that's still the major question in the field. For the moment, I'd just like to stress that we look at the human microbiome from this systematic point of view, or try to look at it in a systematic manner. And just to give you a few notions of what I think is important to look at in the context of the human microbiome, and I'll give you some data on the human gut microbiome. I think it is really important to stress the notion that it is highly diverse. We're typically dealing with hundreds of thousands of strains in individuals. These strains are the ones that form these complex ecological networks that in turn confer essential functions. And what also I think is important, if we're in particular looking at industrialized Western populations, is that we need to be aware that these have already been heavily impacted since the advent of the Industrial Revolution. So getting to the point of actually understanding mechanisms, I think just like to take this kind of bird's eye view and just to highlight a few commonalities in terms of the studies and what we have learned over the last decade and a half. You have probably seen diagrams of this nature a few times before. What we do see is that in the context of disease, there is an apparent imbalance in particular the gut microbiome. And this is oftentimes referred to as dysbiosis. And then in contrast to that dysbiotic state, we would have a eubiotic state associated with health. And so it's oftentimes portrayed in terms of this balance. But what I would like to stress is if this is indeed the case, and it does appear so from the innumerous papers in the context of different case control studies on different diseases that have been done so far, that also implies, of course, that there is a different biomolecular spectrum that would be produced in the context of dysbiosis, and then, of course, the interface with the host. So this apparent dysbiotic state has been reported for many different chronic diseases, ranging from autoimmune diseases to neurodegenerative diseases. But then I think in terms of thinking about causal consequence and mechanisms in particular, what is also important to stress is, of course, that on the host side, we have a similar apparent imbalance in terms of the immune system. And this is another unifying factor in that in the immune system of diseases, we have an apparent imbalance, so typically referred to, of course, as inflammation. And so the key question here is, of course, over chicken or egg, cause or consequence. And I think it is important to note that this will be complex, and it is therefore important that we apply the vast repertoire of methods to this problem. So we have, over the course of the last decade and a half, developed a number of methods that were built on the methods that Jill, of course, developed. And so what we do is that we take samples, we immediately snap freeze these, whether these are environmental samples or whether they're actually human microbiome samples. We have developed a cryogenic cell lysis that is indiscriminate, and then from a given single sample, we're able to obtain biomolecules ranging from DNA to small molecules in sufficient quality and quantity for downstream multi-omic analysis. We have then developed the necessary in silico workflows that rely, for example, on the reconstruction of metagenome assembled genomes, which then, in fact, represent a template for data integration. In the context of also the talks from this morning, I wanted to highlight one tool that we have developed in this context also, that is PathoFact, that is for the prediction of virulence factors, toxins, antimicrobial resistance genes, and also biosynthetic gene clusters. And what is also unique about PathoFact, it places that into the relevant genomic context. Important also to stress is, in particular, based on the cell lysis method, is that we, from a given sample, can, of course, obtain basically data on bacteria, gram-positives, gram-negatives, archaea, microeukaryotes, as well as viruses. And more recently, we have taken this approach of generating integrated multi-omic data from a given sample, and we've expanded it because what I've highlighted earlier is that what forms the interface with the host from the microbiome is this cocktail of biomolecules. And the cocktails that are relevant here are, of course, mostly in relation to what is extracellular to the microbial cells, for example, in the gut. So along with looking and systematically measuring intracellular DNA and all of the other biomolecular complements, we're also extracting now extracellular DNA, extracellular RNA, extracellular proteins, and extracellular metabolites along with the intracellular biomolecules. And so why is that relevant and why is that important? Well, I have already introduced this notion that the interface with the host is through the extracellular biomolecular complements, but this is just pilot data from the initial basic methods paper that we did, and this is for 4 healthy individuals. And in a metagenomic typical analysis that you would do, you would just resolve intracellular DNA, which is always on the far left of these bar charts. And what is important to stress is that based on the information in the intracellular DNA, you, of course, cannot necessarily predict what would be mirrored outside of the cell. And so I think it is important to measure these different biomolecular complements, because each and every one of them may in particular be relevant from what I mentioned earlier, and that is from the point of view of inflammation and the interface with the human immune system. And so this is again just the pilot data from our methods paper, but what is clear is that in each of the different biomolecular complements, what you see are immunogenic molecules, such as we found, for example, that the extracellular DNA and extracellular large RNA is enriched in CpG motifs, which are relevant for the binding of toll-like receptor 9, and then the production, for example, of AL8 by macrophages. And you see this along all of the different biomolecular complements. And what is also crucial is that you see enrichments from certain taxa in some of these complements, such as, for example, in one individual in this very small study, of course, we found that the extracellular large RNA was dominated or was actually overrepresented by RNA from Methanobrevibacter smith from the dominant archaeon in the gut, where we found that 5% of reads were originating from that particular taxon. So one thing is the measurement, but then there is already, of course, quite a lot known in relation to the biomolecules and how they then interface in particular with the human immune system. And to just get an overview of what we know already, we basically built this ExpoBiome map knowledge graph, which I'm not going to take you through in detail. I just want to highlight that what we have in this are basically individual microbial taxa, biomolecules that they produce, and then how those biomolecules indeed would trigger immune pathways, and then linking these to diseases where those relevant immune pathways would be perturbed. So this is, in fact, a knowledge graph that is based on tax mining and manual creation of access of 17,000 individual peer-reviewed publications. And it's important for us to have this as a sort of baseline onto which we can map data and see where the data that we measure agrees with what is known. But then importantly, this is also a means of identifying novel molecules and, of course, potentially novel pathways. And the challenge here is actually quite daunting because what we know from the measurement side is that in a given individual, we're dealing with millions of distinct biomolecules, again, ranging from nucleic acid moieties to small molecules. And if we look at the mapping of those biomolecules in terms of the measurements against the existing knowledge, the vast majority of those biomolecules are unknown, and they're also unknown with respect to the impact that they have on the human host. So they are, for example, unknown with respect to their immunogenicity. So I'll give you an example of a class of diseases that we are currently working on. I had mentioned earlier the whole panoply of different human chronic diseases, and one particular class of chronic human diseases that is now linked also to the gut microbiome is neurodegeneration. And more specifically, we've been working on Parkinson's disease, which was also mentioned in the earlier talk, and that in the context of genetic forms of Parkinson's. And what we are specifically focusing on in our work here on the microbiome are idiopathic forms of Parkinson's, so for which there is no genetic basis. So how might the gut microbiome trigger processes that would lead to neurodegeneration, and more specifically, Parkinson's disease? So the gut microbiome may indeed produce an array of different biomolecules, and one particular example of biomolecules that it may produce are actually amyloidogenic proteins. So these are proteins that form amyloids, and a common denominator in relation to neurodegenerative diseases are also these aggregates of human proteins, in particular neuronal cell populations. So those, in the context of Parkinson's, that would indeed be dopaminergic neurons, for example, where you then have aggregates of the human protein alpha-synuclein. And so what has been postulated is that, and I'll give you some data on this, this is actually also the case from experimental models, is that amyloidogenic proteins from the gut microbiome may actually seed amyloids on the human front, such as alpha-synuclein. Then you get aggregation of those proteins in the enteric nervous system, and then via the vagus nerve, from one neuron to the other, in a prion-like manner, you get basically the spreading of these aggregates, such that you have then the first hit in terms of the central nervous system would be the brain stem in the context of Parkinson's disease, and that is also based on very intricate neurodissections done by the famous neuropathologist Heiko Brack in Germany. Seems to also be the case, and only at later stages of the disease are then other susceptible brain regions affected, and only really very late is, for example, the cortex affected. Another route might, of course, be also via circulation. And so it's just important to recognize that now a lot of work has been done on different neurological conditions, and common denominators appear again to be that you have a microbiome, in particular gut microbiome shifts in the context of these neurological disorders. And I wanted to just walk you through some of the results here, and then get to a more recent study, and then, of course, conclude on how we can now test hypotheses in relation to this. So this is from our earliest work on the gut microbiome in Parkinson's disease. So we here looked at the gut. We also looked at the nasal cavity, and importantly, we looked at individuals that have a prodrome of Parkinson's disease. And this prodrome is idiopathic REM sleep behavior disorder. So people with that condition who act out dreams basically have a 90% risk of developing some form of Parkinsonian-like condition in the subsequent 10 years. And what we had seen in the prodrome, but more importantly in relation to newly diagnosed Parkinson's disease patients, are indeed shifts in the gut microbiome. And we were first to report that, in particular, in certain individuals with Parkinson's disease, you see that you have an enrichment in the genus Akkermansia. The type strain is Akkermansia muciniphylla, and as that name suggests, that taxon in the varicle microbia actually likes mucin. It's a mucus foraging bacterium. And what is also crucial from this study is that for certain individuals you have elevated levels for Akkermansia, but then for others you have other taxa that have similar functions that are also mucinolytic or mucus foraging to be elevated, such as, for example, Bacteroides. And this has now been replicated by hundreds, actually, of studies internationally, and has also been, for example, nicely summarized here by Rosario from a functional point of view, and that is that in the context of Parkinson's disease, we have an enrichment in genes generally for mucin and host glycan degradation, while at the same time we have a reduction in starch and other polysaccharide hydrolyzing enzymes. And so this enrichment in mucinolytic organisms was really interesting to us, because we had actually performed a completely unrelated study together with Eric Martins at the University of Michigan, where we had aimed to compare fiber-rich versus fiber-deprived diets. This is work that was led by Mahesh Desai from India, in fact, former postdoc in our lab. And so when we compared the fiber-free to the fiber-rich diet, and this was a synthetic community of microorganisms, what we saw in particular in the fiber-deprived diet was an enrichment then in Akkermansia muciniphylla, along with other mucinolytic bacteria, such as members of the Bacteroides. And what that leads to in vivo is that you get basically an erosion of the outer mucus layer, a thinning thereof, and what that results in particular then in the animals that have this eroded mucus layer is that they have a much higher susceptibility to infection by an enteric pathogen. So our hypothesis here was then based on the findings in Parkinson's that this enrichment in mucus foraging taxa may then basically erode away the mucus layer to a large extent, and that would increase their propensity to be exposed to a given toxin that may be triggering then the disease processes along the lines of what I showed earlier. So that was our hypothesis. And so what might such a toxic molecule be? So it is well known that, of course, different taxa, particularly in the gut microbiome, produce amyloidogenic proteins, mostly, for example, for biofilm structural properties that they confer. And so this is some work from the lab of Sarkis Masmanian at Caltech. They've been working on this also for a while. And so what they showed, and we have very similar results actually, that is that basically CSGA, which is one of the subunits of Curly, that this can act as the template for the misfolding and subsequent aggregation of human alpha-synuclein. Again, alpha-synuclein is the classic molecular hallmark of Parkinson's disease in that you have aggregates of it in dopaminergic neurons. Those are the neuronal cells that are mostly affected. Okay, so basically a bacterial protein can act as a template for the misfolding of the human protein. And more recently, I've become very interested in other amyloidogenic proteins, in particular small proteins. If anybody's interested in that, we'd be happy to discuss. But so in a follow-up study done by Chris Schmitz in the lab, we were then looking at manipulating 3 factors, and that in a murine model. The first was basically we had wild-type mice and then a transgenic mouse line overexpressing human alpha-synuclein. We modulated the fiber content in the diet, low versus high fiber diet, and then on top of it, we exposed those mice to either a Curly expressing or a knockout strain of E. coli. And so what you already can gather here is that we're, of course, in a multi-dimensional space here. I'd like to just quickly summarize what we then found with changing those 3 main factors was what we saw very much along the lines of what Mahesh had observed in his initial study was that the fiber-deprived diet leads to a change in the gut microbiome, leading to dysbiosis. That has then an impact on the gut barrier integrity. And if you, on top of that, add the insult of exposing the mice to Curly, so again, this amyloidogenic protein, you get more aggregation of alpha-synuclein in the enteric nervous system, in enteric neurons. This then propagates to the central nervous system, where again you have more aggregates form, and you have the classical hallmarks of degeneration, which then manifests itself in motor deficits, where there was a trend to the classical hallmarks of Parkinson's disease in these mice, but didn't reach statistical significance. So one would have to, in fact, repeat this study with a larger cohort of mice and also follow them over a longer time. But just to conclude here on this part, what this shows is that microbiome-derived molecules can be a trigger in the context of known disease pathways, and this in the context of Parkinson's disease. So one thing is to come up with mechanistic study. A whole other kettle of fish is, of course, in relation to the different factors that might be involved is measurement. So just to highlight here a more recent study that we've now done on actual, again, patients, and this is an integrated multi-omic analysis of basically fecal samples from around 50 controls, 50 Parkinson's disease patients, and 30 individuals with idiopathic REM sleep behavior disorder. Again, that's that prodrome of Parkinson's disease. And so what we generated is metagenomic data, metatranscriptomic data, metaproteomic data, alongside metabolomic data. And so we have ways of course, by now integrating this data. And what is important in our reinterpretation here of any mass transit system is that at each junction, you can extract different information. So you can look at whether, for example, comparing only metatranscriptome to metagenome, whether you then see differences of certain enrichments in one condition versus the other. And so we can then systematically go through this data. We've also done this for other diseases, including colorectal cancer, type 1 diabetes, and the learnings actually are very, very similar. So what we see for the metagenomic data is that there are no apparent differences between healthy and diseased individuals, and we also see no apparent functional differences. And so this Permanova analysis gives you an indication for the fact that we can then compare all of the different data sets, and we can also relate those to potential confounders. So basically effects that one would see in the microbiome that might be due to, for example, other hallmarks of the disease, such as constipation or indeed sex. But what this told us is, in fact, that basically where we see clear discrimination and clear discriminative signatures is, in fact, in the metatranscriptome of gene functions, as well as in the metabolomic data. And these really represent the most discriminant omic data sets. We then focused on the metabolomic data because the metabolomic data reflects, of course, the closest to community-wide phenotype. And as you also see from the Permanova analysis here on the right-hand side, it is actually the least affected by the confounders. So here are examples of 11 metabolites for which we found then statistically significant differences between healthy and diseased individuals. You see that there is a clear signature here for example, amino acids. But important also in this is that we can then try to start relating those metabolites to constituent taxa and to constituent functions. And for the sake of the length of today's talk, I will not go into all of these details. I will just give one concrete example, and that is for beta-glutamate. So beta-glutamate was first described as an archaeal osmolyte, and you see that it's the most statistically significantly correlated actually with the genus Methanobrevibacter. Also in relation to the taxonomic ranks, we can of course resolve this data at the species or strain level, but I've summarized again for the sake of simplicity here to the genus level. And so what you see is that there's this strong association with Methanobrevibacter and beta-glutamate. Beta-glutamate is very interesting because hardly nothing is known about it. You will recognize L-glutamate, and in particular its enantiomer D-glutamate. So for them, for those two metabolites, a lot is known in terms of pathways that they feed into, but for beta-glutamate, that is not the case. It's really, really important to figure this out because L-glutamate is the most important excitatory neurotransmitter, and a lot is seen to be affected in relation to neurotransmission driven by glutamate in Parkinson's disease, in particular excitotoxic effects. So again, digging a little bit here into the data, what we see is that, for example, beta-glutamate, again, this is this archaeal osmolyte, is negatively correlated with glutamate synthase, so you get less glutamate synthase, which is the key enzyme involved in the synthesis of L-glutamate, which again is the excitatory neurotransmitter. And at the same time, what we see is a reduction in glutamate-linked chemotaxis, which is also interesting in relation to the first talk of this morning. So there are many knock-on effects that one sees related to glutamate. And I mentioned already glutamate-linked chemotaxis. So what we see is a reduction in glutamate-linked chemotaxis in the context of REM-sleep behavior disorder, but importantly also in Parkinson's. And that actually then also impacts flagellar biosynthesis. And also very crucially, what we see is that there's a clear reduction in the diversity of expressed key functional genes such as glutamate synthase. And so this is a very busy diagram. I'm very much aware of this, but it basically, if we just focus on the flagella, it basically reflects what I've just told you in a nutshell, and that is that in relation to flagella as well as chemotaxis pathways, we have in apparent 4 distinct clusters comparing, again, healthy to diseased individuals. So for the vast majority of these genes in terms of their expression, there's more or less even expression between healthy and diseased individuals. But then for Parkinson's disease, as well as for healthy individuals at the bottom, you see that there's almost exclusivity in the expression of certain flagella genes in particular. And so this is really interesting, not least in the context of the inflammatory dimension that one sees in Parkinson's disease, where you have clear neuroinflammation, and also in relation to more recent work by Ruth Ley, who's described these silent flagellants that have apparent anti-inflammatory effects or neutral effects. And so the question here, of course, is whether some of these flagellins in particular may be more pro or anti-inflammatory. And so this is just, in essence, a summary of the functional findings that we have linking different changes at the level of expression of different taxa to functional pathways, and then how those impact host physiology, again, in the context here of Parkinson's disease. And of course, here I've just focused on glutamate-linked chemotaxis, and then flagellar biosynthesis in particular involving flagellin. And that just gives you an idea of the fact that we can now move through these different landscapes in relation to molecules, link them to functional pathways, and really understand what is going on, what might be a driver, what might be a passenger, and importantly, what might be a trigger, in fact, of disease. And so that brings me just to the end in just highlighting the fact that we are dealing here with many, many, many different factors, of course. So these are very complex diseases that only develop, over the course of probably 20 years that we know by now. And so we need to have models by which we can then test all of these different, basically multifactorial hypotheses. And one system that we have built, there are also now others being, of course, engineered, is our humic system. So this allows for human microbial co-culture. And most recent innovations are that we are able in this system to co-culture with human cells, strict anaerobes. We can monitor, for example, barrier function online to assess what the effect, for example, of mucinolytic taxa might be on the barrier. We've also recently succeeded in culturing linearized gut organoids along with enteric neurons in the system, and we've more recently have also been able to culture human PBMCs, so immune cells in the system, and we've interfaced it with other organs on chip. Okay, so this brings me to the conclusion. So I've given you a notion of the wet lab and dry lab methods that we have developed for systematic integrated multi-omic analysis of microbial communities. So if we stick to the term expobiome, just for the sake of framing this properly, this of course represents a cocktail, a complex cocktail of biomolecules that may trigger these complex processes that culminate in human chronic diseases, and that might be then the basis to think about if there is any such thing as pathogenic microbiomes. What we have continuously seen, and I've given you the concrete example for Parkinson's, is that there's no real taxonomic differences between healthy and diseased individuals. Yes, you might have certain taxa that are up in certain individuals, but then that's not the case for other individuals with a given condition, and so that means that there's clear functional disruptions in the context of chronic diseases, and that means also that I think we should really focus on what impacts us as the host, and that is function, in essence, that is conferred by the microbiome. And in order to test all of these hypotheses that arise from this data, we really need new high-throughput methods to do co-culture, such as with humics. And ultimately, what we are aiming here is to unravel cause and consequence relationships. I'd just like to highlight, if any of you in the audience are thinking about doing a PhD, we just have received funding late last year for a new doctoral training unit entitled Pathogenesis in the Age of the Microbiome, where we will be tackling a lot of these questions. So there's 16 PhD positions available. I'm the coordinator of this program, so if any of you are interested, please come and see me. A lot of people to thank with this, and then, of course, thanks again for your attention, and happy to discuss.
Thank you, Dr. Williams, for the excellent talk. Any questions? So there's already.
That was really awesome. A question, how frequently does this metabolite composition changes over time? And, by the impact of this fiber diet and the non-fiber diet, when does the impact tends to go away? Like, does the microbiome comes back and stabilizes? So if you give an insult of a non-fiber diet and then brings to the fiber diet, does the microbiome go back to its original form?
Okay, I'll tackle your second question first. So basically, if we switch the mice from a low fiber to a high fiber, basically in the context of the synthetic community, basically you have a reset virtually, over one day, right? So it changes very rapidly. I mean, at the frequency that we can measure this, right, without euthanizing the mice. So we just collect fecal pellets in the context of this work, because we want to limit animal experiments in any case as much as possible, but they're indispensable for some questions. So yeah, that is one point. And then in relation to your first question, generally I think the fiber is obviously a driver in relation to many, many functions, not least in relation to short-chain fatty acids that have other physiological effects. So I don't know if that answers your first question as well. Okay.
Brilliant talk, Lance. Thank you. So I actually had a question that you mentioned that deprival of mucin is probably the reason why the toxins are exported, right? Well, so intervening this right from the early time, like if you have a mucin supplement or food that would be highly mucin rich, would that help in preventing the disease, or do you think that will help in the progression of disease would be stopped? Or
yeah, no, I think actually basically including mucin in the diet would probably have the opposite effect, because you would enrich more for mucinolytic taxa. What I think is clearly the advice that comes out of this in relation to nutrition is that a high-fiber diet appears to be protective against mucinolytic bacteria. And also, these findings have also been replicated now in a number of other studies. So I think that is the key message, that a diet rich in fiber basically enriches in taxa that are capable of hydrolysis of the complex carbohydrates in fiber, rather than feeding off the mucin and mucus in general. Yeah.
One question.
Hi, Paul. Great talk. Really enjoyed it. Thank you. I have one methodological question and one general question. For the methodological question is, how do you account for all these different factors, comorbidities, medications, in building the cohorts between disease and control to account for everything that influences the microbiome? And second is, which is a general question, have you looked at the oral microbiome at all?
Yes. Okay, tackle your first question first again. Yes, we look at the oral microbiome. We have actually published quite a lot on this now, also in particular looking at transmission from oral to gut. We've looked at that in the context of colorectal cancer, of course, because in colorectal cancer, on tumor as well as in feces, you have an enrichment in facultative oral pathogens. But we also see very interesting patterns, for example, in type 1 diabetes, where you have a transfer of more pro-inflammatory streptococci from the oral cavity to the large intestine. That also, at least associate with some of the inflammatory markers. Sorry, and then the first question was... sorry?
The methodological association with comorbidities medication.
Sure, of course, yeah. So what is important is that we have a lot of, we define, of course, quite a lot of exclusion criteria when we select our patient cohorts, but also very important is that, for example, especially in the context of Parkinson's disease, so these are de novo diagnosed. So they have not yet received any anti-Parkinson medication. That is really crucial. And we have the prodrome. I think we have to do much more about prodromes in microbiome studies, because like in particular neurodegenerative diseases, whether it's Alzheimer's or Parkinson's, the original insult probably occurs 20 years before you have manifest symptoms. And if the microbiome is somehow a driver there, it's possible that what we see at the point even of diagnosis, does not any longer reflect the original insult.
In this non-genetic...
Please go ahead.
In this non-genetic version of Parkinson's disease, do you also see alternative lines of evidence like high fecal calprotectin, and also do you see comorbidity? Because you are proposing a very strong dysbiosis, and then that is causing the toxins, which is perhaps causing Parkinson. So I wonder if you see other lines of evidence to support this theory.
Yes, basically in relation to calprotectin, yes, you have elevated levels of calprotectin typically in the stool samples of Parkinson's disease patients. You have other manifestations of gut barrier deficits. And so, yes, I agree that, there are many hallmarks here that point to the gut, not least, I mean, the original link was done around the notion that around 30% of Parkinson's disease patients have constipation. So there is already a deficit in relation to the gut. And so there are a number of lines of evidence, not least also the one that I didn't stress so much on, what was found about 10 years ago by Pascal de Kindan in Nantes in France, is that you have already, before people develop manifest Parkinson's, you have a lot of these Lewy body inclusions, so basically aggregates of alpha-synuclein in enteric neurons. So that really points to the fact that, at least in some cases of idiopathic Parkinson's disease, what happens in the gut happens early and hits the central nervous system later.
We'll take one last one from the audience.
And I think it's a very short one. So everyone would like to do multi-omics. I think we are very sample limited in environmental regions. So just wanted to check with fecal samples for this multi-omic analysis, how much sample you needed for this work, and how would it translate to any sort of environmental sample?
So
they are low diversity.
How it relates to environment?
As in the human fecal samples are very rich and you have much more stuff compared to environmental samples where there's less diversity. How would it translate the same thing?
So we have been successful in doing this on 10 to the 7, 10 to the 8 cells. Also from environmental samples. For human fecal samples, we typically work with 150 milligrams of stool. We also, I mean, we've worked a lot. In fact, the whole methodology we developed on biological wastewater treatment systems, and there also we deal with 100 to 150 milligrams of sample material. That depends there again on the cell count. But we can by now go rather low.
We'll take one last one from the online. So the bottom one, can we consider microbiome dysbiosis as one of the potential factor contributing to the dysfunctional immunity in microgravity?
I mean, that's possible. I don't know if there are major shifts I assume, in the microbiome, given the diet also of astronauts, etc., that would be in microgravity environments, if it's like, for example, in the International Space Station. So that potentially could be indeed a driver if inflammation is a concern there, for sure. Yes.
Great. Thanks a lot, Dr. Williams. Yeah.
Just a quick announcement for the international speakers. We have a meeting with our Dean of Global Engagement now, so please do join us. We have a short ride. It's just the adjoining building. So please meet us outside in like about 5 minutes. Thanks.
No, we have flash talk, then tea break. You can switch.
This is not there on the website at all.
Yeah, it's not on the website. This is.
So there is no flash talks after this.
Yeah, because even I found out it was tea and this one. So. Okay. And we'll put flash talks at 12 to 12:30 flash talks. We'll break for tea now, along with the poster session 5, and at 12 we'll have the flash talk session.
Rajan. He is currently Associate Director and Senior Group Leader in the Genome Institute of Singapore. Now, his lab mainly focuses on the development of genomic analytics tools for studying the function of diverse microbial communities and their impact on human health. Now, he is here to present the tackling the global spread of AMR using genome-resolved metagenomics and AI. So, please.
Okay, can you hear me now? Okay, great. Okay, as all the speakers have done, thanks to Karthik and Karthik. It's really a pleasure to be here. I think it's been a decade since I came back to IIT Madras to give a talk. So again, it's a great opportunity. It's my hometown in some sense, so it's good to be here. But on another note, I think it's great to have a conference like this in India focused on the microbiome. I think there's a greater need for this to make this an annual thing. I joke with some of my colleagues in Singapore that we meet each other more in somewhere in Europe or in the US for a microbiome meeting rather than in our own cities and neighborhoods. So it's nice to have more such meetings in South and Southeast Asia, I think. Okay, so let me get started. Ignore the high-sounding title. I'll get into what we are really trying to do, if I can actually move the pointer. That's a problem. This is not connected. Give me a sec. Okay, so this figure has been shown multiple times in this conference, and it's really to highlight the fact that a lot of biological research is focused on humans for me, and that really represents a tiny sliver in the genetic diversity of life. Yeah, and that's really, unfortunately, because microbes are so tiny, hard to see, we don't appreciate how much of life is microbial. It's estimated to be between 1 million and 1 trillion species out there, of which less than 100,000 for which we have high-quality genomic information, and only a few thousand that we actually have isolates for. So perhaps this is one of the situations where there's a lot more that's unknown, and there was this mention of India's population. If each of us went out there and tried to isolate our own unique strain, it's there for us to isolate potentially and characterize it, and we can all name a microbe after ourselves if we wanted to, really, right? So there's that much life out there to be studied, and yet humanity's interaction and perception about microbes has been shaped by the fact that our first encounters have all been, or the ones we paid attention to, were infections. And when we encountered these infections, our response obviously was to kill microbes with various agents, antimicrobials and disinfectants and so on. And that has led to a vicious loop. And others have mentioned the AMR challenge. I'll just flash this old figure which tries to quantify the scale of the problem if we don't address it now. But I think we already have a challenge, especially in India. Perhaps many of you are aware, we have often situations where people get to the hospital, get treated by the first line of antibiotics, and the infection doesn't go away, and then there's the real fear that we might lose a loved one. So I think this is a problem we feel here every day, and to some extent in Singapore as well. And so my lab is very interested in seeing how metagenomics can attack this problem, not from developing new antibiotics. I think that's a very important area, but we're trying to kind of rebalance our understanding of the microbial world, not just focus on the pathogens, but also the diversity of species which are not pathogens. So we are really focused on technologies. I'm a computational biologist by training, but my lab works on experimental approaches as well. And this is to improve our ability to look at microbial ecosystems around the world. We think part of our challenges is we're not able to observe well, we're not able to isolate well, we're not able to model microbial interactions well, and we want to increase the amount of data we have and the quality of models we can build. We are doing a lot of work on skin microbiome. We see that as an important part of this picture because skin is an important interface between the environment and the human body. But also it plays a role, skin microbes play a role in various common skin diseases like eczema, and so that's an area of focus. I'll say more about our work on antibiotics and gut microbiome. Karthik alluded to it. But here we are looking at how antibiotics perturb gut microbial ecology, how that recovers, and how colonization resistance potentially plays a role in risk for colonization by various pathogens, where the lack of colonization plays a role. And the last piece is we are interested in being able to use metagenomics for environmental surveys. We have done some work in hospital environments. Recently, we worked on hawker centers, which are areas where a very large fraction of Singapore's population gets food daily, so they don't cook at home. They go out and eat every day, but they are these very efficient, cheap hawker centers which provide a diversity of food, but it also potentially could be a hub for foodborne infections and outbreaks. So we think this is important to be able to showcase the power of metagenomics for surveillance, and so we have done studies in this. And of course, we are part of MetaSub. It's been a great experience being part of MetaSub. Okay, so what do I mean by metagenomic surveillance? Yeah, traditional surveillance is defined by what clinicians do, and their main goal is to say a pathogen is there or not there. Yeah, and to some extent, they might go further and say there's a particular serotype in there, or they say there's certain antibiotic resistance gene is found, therefore there's a particular phenotype there. But primarily, it's high confidence detection of pathogens. Yeah, and so there's diverse workflows, one for each pathogen, literally. And of course, you can see the value of metagenomics. It's a one-shot, one ring to rule them all kind of idea. But of course, the ones we have been typically using, in the field of metagenomics has been using the 16S approaches or the short-read, shotgun metagenomics approaches. They don't quite get you what you want from a genomic surveillance perspective. Because from a genomic surveillance perspective, you want the whole genome of an organism, because that's the unit you want to use for phylogenetics or understanding resistance mechanisms and so on. So a while back, and I know in this conference a lot of people have talked about it, but this is something we were interested in the early 2014s, where long-read sequencing had just started coming, and in particular we were excited about nanopore sequencing as a cost-effective way of doing this in a clinical setting. And of course, then the challenges were the error rates were very high, in the range of 15%, actually I remember data sets which had 30% error rate. So we worked on algorithms for being able to deal with that error rate. But also, can you really use these technologies in clinical samples? And at that point, we were one of the first groups to show that, yes, we can take real stool samples collected in a clinical study and then get high molecular weight DNA and be able to get enough throughput. So we get gigabases of sequence. We get N50s in the 5 to 10 kb range. So this is really useful for assembly algorithms, as I will describe. So we developed an assembler a while back. I won't go into details because to some extent it's a bit outdated now. The field moves really fast in terms of both sequencing technology and assembly tools. But there are still some parts which are valuable. I think we have found some areas where a hybrid assembly workflow that leverages the strengths of short read in terms of the base quality, as well as the strengths of long read to be able to stitch those bases, those short contigs from short reads into longer sequences. That has some value even now in metagenomics. And another interesting part about our method is that it actually leverages a database of reference genomes. So it's not based on that database, but it leverages that. So it's a way for us to continually improve our assembly algorithms based on genomes that we have already acquired. So just a quick view of what we got from this assembly algorithm. This is, again, half a decade ago, but it already shows that instead of having Illumina contigs, which are few kilobases long, we now get hybrid contigs, which are hundreds of kilobases long. So this is a 10x or even higher improvement. And this sort of work can even be done on soil samples. So I'm not showing you these results, but even in complex communities like soil, you can make a hybrid approach like this work, while brute force long-read sequencing may not work. Some results were showing that even with that generation of long-read assemblers, a true metagenomic assembler is what you need to have better contiguity. and we can get strain level resolution in terms of the assembly. Yeah? So this is very important for clinical studies because let's say you're looking at a stool microbiome, you might have 2 strains of E. coli. One of them is a multi-drug resistant one, and the other is just the commensal that's hanging around. And so you want to be able to distinguish those two when you assemble those genomes. Okay, so just a quick vignette about what we got out of this assembly. I know some of you are interested in plasmids and phages. So we were initially just looking at the circular contigs, and we get a range of contig size from a few kilobases to hundreds of kilobases. So we're quite excited to see what these were. As it turned out, the ones which were very long also tended to cluster here. They had no identity to anything in the database. Very little of them was covered with blast hits. And one of the things we found we were quite excited about at that point of time because we annotated it, we found all the relevant phage proteins. In terms of quality, assembly quality, we could see that the long reads were tiling that sequence completely, so we didn't have to, we tried to curate it, but if we didn't have to fix anything, there were not even base error-wise, we couldn't find clear signatures of base errors. And so this, we believe, is perhaps for the one of the first evidence of a fully automated reconstruction of a jumbo phage from the human gut microbiome. So, of course, by now, again, if you have hi-fi data, you can probably even do more. And this is becoming easier and easier, but I think the opportunity for discovery is there. Okay, so I'm going to switch to a practical application. And we challenged ourselves saying, hey, what can we do in terms of clinical applications? So in this case, we were looking at hospital environments. We had some clinical collaborators who were willing to take the jump because infection control in hospitals is primarily focused on patient-to-patient transmission. That is what gets people worried because there's one individual who in the hospital who had an infection, and a few days later, another individual who in the same ward got the same infection, and then they start suspecting there was a transmission happening, and then you might have to shut down the ward or something like that. Let's say if it's a neonatal ICU. So patient-to-patient transmission is focused on, but nobody looks at the environment. It's convenient to ignore it because there are so many environmental services that harbor microbes despite best cleaning measures. And so if there is transmission, almost certainly there's transmission, they do not have the bandwidth to do a surveillance study on that. So we said, hey, don't worry, we have the technology. Let's see what metagenomics can do, at least genome-resolved metagenomics can do. Again, this is quite old by now. But what we did was took some high-touch surfaces in the hospital environment. We used a combination of Illumina and nanopore metagenomics to profile them. And one of the interesting things we did here was for some of the swabs, we actually plated them. So we actually culture them for a while, and perhaps with even with different media, with different antibiotics. So now what we're getting out of the metagenomics data is sort of a quasi-metagenomics where we have some evidence that these bacteria are alive at the point they hit the plate. And then we also have a sense of their resistance phenotype. And with a small number of swabs, a few hundred, we get thousands of high-quality metagenome assembled genomes, closed plasmids, thousands of plasmids, each of which has N15, the megabase pair range. So the assembly has assembled the genomes to a few large contigs. And so now you can do genomic surveillance, true genomic surveillance for a large number of species. This is something that's, even to date, there are no other studies which have done something like this, a multi-species kind of surveillance in a hospital environment. I'll just show you what you can learn from this. So this is Staph aureus, a common pathogen of concern in hospitals. We took all the Staph aureus genomes, we clustered them at the 99.9% identity. So this is to reduce the information. We constructed a phylogenetic tree, a rough one, not a high resolution one. And then we highlighted the resistance patterns. And you can find that there are some strains which seem to be highly resistant, and at least to the 5 different classes we tested. And then it seems like these same strains also tend to be widely disseminated in the hospital environment. So despite whether it was an isolation ward or a multi-drug resistant ward or different sites of the hospital environment, we find the same multi-drug resistant strain is disseminated. So, of course, you can say, well, this is just this is a nice vignette, but is this a real pattern? We can actually do statistical testing, and we can actually show that statistically speaking, the more multi-drug resistant you are, the more likely you're widely disseminated and persistent. So here, the two colors are 2 time points 1 week apart. So we just have some sort of local stability of the strains in the hospital environment. We also had a 3rd time point a year later, and we can show similar patterns in that. And you'll say, well, this is just one species, maybe Staph aureus is bizarre, and this is where the metagenomic surveillance aspect is nice. We can show the same pattern again and again across species, that the more multi-drug resistant you are, the more widely disseminated you are in the hospital environment. Okay, so we thought, well, that's interesting, and we were hoping to now compare this to isolates or strains from patients, but we did not have IRB for doing that. But we had the opportunity to compare against historical data. So in this case, this is data collected about 8 years ago from the same hospital. It was an MRSA surveillance study. So we had genomes from skin swabs from patients in the hospital, and we can compare that to our genomes. And we did not expect to see any hits because this is 8 years apart, but we had the data, so we looked at it. And it turned out we could find hits. We found hits which had less than 10 SNPs in the whole genome between these 2 strains, and these strains are temporally spaced by 8 years or more. And interestingly, again, we found this strain S3 that we marked in the previous figure. This is the one which is resistant to many different antibiotics, but is also seen in the patient. Yeah? So this made us test this hypothesis. Are more multi-drug resistant strains also enrich the intersection between the environment and patients? And lo and behold, that's what we found. And not only for a single species, but for multiple species. So we seem to see this pattern of hospitals being enriched for multi-drug resistant bacteria, even though we would hope that it's exactly the opposite. We don't fully know why this happens. We were hoping to do a longitudinal study in the early 2020s, and then COVID happened. But nevertheless, I think this is something that can inform infection control, hopefully catch outbreaks even before they happen. Okay, I'll switch gears now to the gut microbiome. So in the context of the gut microbiome, it's another important place to do surveillance in the sense that you might be colonized by multi-drug resistant bacteria, and then the concern is, even though it's just a colonization, it's not an infection, there might be exchange of plasmids across bacteria, and that might transmit antibiotic resistance as a phenotype across bacteria, and then you can have a silent transmission happening through the population where through the oral fecal route or through other modes, you're getting this resistance mechanisms being transmitted to different individuals, and perhaps even to things which are not originally pathogens, but now are all multi-drug resistant, and so a chance infection, chance opportunity to be in the bloodstream or something can lead to a life-threatening infection. So in this context, we did a study where we had a clinician who was willing to collect stool samples for us. So this is of people who are taking antibiotics. So this is not a control study. These are just people who are taking antibiotics. We approach them, we collect fecal samples, and we do metagenomics longitudinally. And it was a very messy cohort because some people will say, well, I don't want to give the 5th sample that you wanted me to collect. I can only give you the first 3. I don't want to give any more stool samples, right? But we had a very resourceful postdoc at that point of time, Tarini, who's now an assistant professor at IIT Delhi, who said, well, even though the cohort is messy, let's try to use this and see a particular pattern, which is this idea of microbiome recovery. So it has been described by David Relman and many other groups that recovery of the microbiome, the diversity of the microbiome post-antibiotics is not a uniform phenomena. Some individuals recover their microbial community diversity within a few weeks, and some individuals will never recover. As in, even after a year, their community looks highly dysbiotic. It has a few species highly dominant in the gut microbiome. But why does this happen? Is it just a pure repopulation effect, like some bugs survive the antibiotics and those guys repopulate? Or is there something more interesting happening in the community? So it turned out that, of course, our data sets were small, so Tarini was clever enough to say, let's get the world's data set in-house and let's just analyze it. Each cohort is small, so we can analyze them separately, but we'll integrate signals across cohorts. And based on that, he found a small list of about 20 species. These species are actually consistent in terms of their functions. There are some Bacteroides species, some bifidobacterial species. And we were able to build several hypotheses, including with Arthi, who was then in Karthik's lab, about how these species might be keystone species. They might be playing a role in terms of cross-feeding other bacteria and thus leading to the repopulation of the microbiome. And so their presence is essential. But we also wanted to test it in a mouse model. So we went ahead and did this very small scale study in mice where we gave them antibiotics and we monitored them. We gave them various bacteria that we were interested in. We called them recovery-associated bacteria. So Bacteroides theta micron, which is a keystone gut commensal, as well as Bifidobacterium adolescentis. It's a common probiotic. And then we used a combination of the two species as well and monitored the gut microbiome for a while. And then we looked at various parameters of the gut microbiome. So in particular, we looked at bacterial biomass. So this is initially during antibiotics, of course, there's suppression, and then post-antibiotic recovery. And what surprised us really was that there was an effect. Firstly, we were not hoping this was meant to be a pilot study, actually, but also not just that, the strength of the effect. So a few days after antibiotic has been stopped, the Bacteroides theta iodomicron group has a 100-fold improvement in its biomass recovery. So it's a rapid repopulation of the gut microbiome, as opposed to the Bifidobacterium alone or the control group. And we were surprised that the Bifidobacterium did not have an effect because it's a common probiotic. Some of you probably have taken antibiotics and probiotics right after, hoping it helps you. But it doesn't help in terms of biomass recovery. But it does seem to help in terms of recovery of the diversity of the community, different species and their distribution in the community. It seems like adding Bifidobacteria to Bacteroides theta iodomicron has a synergistic effect. So this really brought to mind for us the idea that we need to look at microbial interactions and how they can cooperate for various phenotypes that we observe. So I want to quickly tell you a little bit about this study because this is moving us closer to the clinical translational efforts. Carbapenemus producing Enterobacterialis are really at the top of WHO's list of highest priority pathogens in individuals who are susceptible and who have, say, bloodstream infections. It has a very high mortality, as high as 50%. And so it's really a scary thing to have an infection for, but you can have asymptomatic gut colonization. And so we wanted to understand the factors that lead to colonization or decolonization. Of course, you can't just catch people who are not colonized and then you observe them as they get colonized because it's a very small number of people which have that phenomena. But we noticed from a previous clinical study that within a year, most people who are colonized get decolonized. So we could do the flip study where we recruit people who have been colonized and just track them longitudinally to see what happens in the gut microbiome. So that's the study we did. We were trying to do various kinds of analysis. I'm just going to largely focus on the microbial ecology aspect of it. It's a small study, small number of index subjects and their family members as controls. Okay, so the first thing we realized or noticed was that just at the gross level, these individuals, despite being asymptomatic, have gut microbiomes that are weird. By weird, I mean at the PCOA plot, which many of you have seen, it is shifted. So the red triangles and circles are the CPE positive, the individuals who are colonized by Carbapenemus producing Enterobacterialis. So there's clearly a shift, and there are 4 different clusters and different enrichment patterns. And you can also show on the on the first principal coordinate and coordinate, there is a shift, right? And there's a progressive shift. As you're colonized, you're much more to the left. As you're getting decolonized, you are somewhere in the middle, and the healthy family members are the ones on the right. But despite this very pronounced shift, when you start looking at differentially abundant species, you don't get a long list. You actually get just a handful of species. So in this case, we got about 9 species. Is it 9? It's actually slightly more than 9. But the enriched species are ones you suspect. So Klebsiella pneumoniae, it's an Enterobacterialis. You expect it to be enriched. But the depleted species were striking when we started to look at each one of them and ask what their function is in the gut microbiome. We saw repeated occurrences of functions that are relevant for being immunomodulatory, so anti-inflammatory in some sense. Butyrate production is a classical one, NF-kappa B inactivation, and so the classic bifidobacteria show up in this list. We were also able to, because this is metagenomics data, we were also able to look at the functional pathways, and we saw enrichment of pathways in the colonized state, which are probably pro-inflammatory. So this is things like sulfate reduction, aerobic respiration, and oxidative phosphorylation. So this is all metagenomics, and this is how we published it. Unfortunately, we didn't have, we just had the DNA from the stool samples, not the original stool samples. But we went back prospectively to try to collect more stool samples and ask the question, is there really inflammation in the gut in these individuals? And it turns out it is. It's a strong marker of gut inflammation, can be seen in the form of calprotectin as well as lactoferrin. There's this clear signature that these individuals have something happening in their gut microbiome, in their guts, whether it is driven by the microbiome or something else. Maybe perhaps these individuals are taking multiple rounds of antibiotics, they're in and out of hospitals. There could be many factors, but there's clearly a shift. We also see that the Enterobacterales colonization can be very high. So here on the y-axis, it goes up to 100%. And you can see that some communities are almost all Enterobacterales. If it's white, it means it's some other species. So it's a stacked bar chart here. And each bar chart is a different time point. So for one individual, you can see how their microbiome changes over the period of a year. And you can see clearly that there are some individuals who can just shift between having E. coli high in their gut microbiome to Klebsiella high within just a week or 2 weeks. And it seems to be quite unstable, and we can quantify that the abundance of Enterobacterales varies a lot. And we suspect that this is driven a lot by host inflammatory processes. But what it suggests to us is that it's an unstable state, and decolonization can be triggered with the right perturbations. And finally, the last piece I want to show you is this question of, is there really concern in terms of plasmid sharing? This is a very hard thing to establish definitively because plasmids can be very small. And in this case, what we had was we had whole genomes, we had isolates for these individuals, and we had one individual who had both E. coli and Klebsiella pneumoniae, CRE strains. And we could detect plasmids which are essentially identical in E. coli and Klebsiella pneumoniae. There are 2 plasmids actually, so it was 140 kilobase pair of sequence. Estimated divergence based on some in vitro experiments suggests that these plasmids don't diverge that fast, and so this is likely to be a transfer within about 6 months, which could have happened in the patient's gut. And also we did some conjugation experiments suggesting that these plasmids can be exchanged between E. coli and Klebsiella. So this is not definitive, but some evidence suggesting that there might have been a transfer of this plasmid between E. coli and Klebsiella. So moving forward, we are very interested in being able to take this further in terms of understanding how colonization resistance works. We were able to create an in vitro model. So this is work in Prof. Khan Yun-Hen's lab, where they're able to grow gut bacteria in a reactor, add Klebsiella pneumoniae to it, and see what happens. So what you observe, as expected, is that if you have a full rich community, it does not let Klebsiella pneumoniae grow. But once you perturb it with antibiotics, then Klebsiella pneumoniae can grow well. And so we want to understand the role of different bacterial species. So it's clearly, this is not a host-dependent phenotype. It is actually a phenotype which can be driven by the microbes in terms of colonization resistance, but these interactions are complex. And as Narendra and Karthik mentioned, we are all interested in modeling. We have worked on various methods that take real metagenomic data and create Lotka-Volterra models out of that data. I won't talk about that today, but just summarize that we are really confident that moving forward, genome-resolved metagenomics can actually have clinical applications, especially in the context of surveillance. And so this is what we are trying to spin off, actually, from my lab. We are interested in being able to apply them in hospital environments and understand the source of multi-drug resistant bacteria. In terms of CPE colonization, there are 2 factors. We think there is a host factor nevertheless, but also we want to understand the microbial factors that lead to colonization resistance. So these are the acknowledgements, including my group members, some of our clinical collaborators. I just want to highlight that the AI part of what we're trying to do is really move forward to a point where we create complex in vitro communities with different combinations of species generates sufficient data to be able to fit these models. We think it's maybe naive to assume that Lotka-Volterra explains large complex communities. We may not know the form of the equations that explain the dynamics of these communities, but we feel that we can learn these models in a data-driven manner that can predict dynamics. We don't think there's any magic happening there. With enough data, we should be able to predict patterns. So this is what we're trying to do. This is a recently funded investigative position in Singapore. So if you're interested in these kind of problems, please reach out. And with that, thank you for your attention. Happy to answer questions.
Thank you. The forum is open for the questions.
Hi, Niranjan. Great work. About the dynamics that you showed over the year, did you have dietary data for the participants?
That's a great question. I get asked the same question every time. No, unfortunately, we did not have dietary information in this first study because it's a typical clinical infectious disease study where they were largely interested in transmission between patients and their family members. That was the major motive. The second study that we are initiating now that has detailed dietary information, and we suspect there is an important role of the diet of the individuals. Yeah.
Lovely work, Niranjan. So I had a question about the very last part.
Yes.
So you are trying to figure out whether the existing colonies they determine the engraftment of a pathobiont, right?
Yes.
So and you are seeing if you do antibiotics, flush them out, then it increase the engraftment. I was wondering if you put a strain of Klebsiella, then does it allow for the new strain of Klebsiella to come in? So are you thinking of some sort of those experiments? So let's say you have a commensal version of Klebsiella without AMR. So
yes. No, that's a very good question. In fact, there are already some studies suggesting that if you have other E. coli or other, say, Klebsiella oxytosa, or you have Klebsiella pneumoniae, which is not multi-drug resistant, they will monopolize some of the resources available and therefore not allow this pathogenic Klebsiella pneumoniae, potentially pathogenic Klebsiella pneumoniae. So that's definitely one strategy. But there's also the understanding that there are other mechanisms that come into play. And things like short-chain fatty acid production and how that affects the pH. So what you want is a strategy that works across individuals. I mean, the goal is to go towards a solution that can be worked across the population diversity that you have. So you want to be able to leverage all the strategies. And then, of course, we are playing these games in a reactor community, and maybe engraftment is much easier. But in the human gut microbiome, there'll be differences in engraftment across individuals. So we think this ultimately a multi-species community is the way forward. But exactly which multi-species community will work for a large number of individuals and community types is something that we would like to figure out.
In the bioreactor, in the first case, did you have a Klebsiella where it was not aligned the new Klebsiella to?
No, there is no Klebsiella in the initial. Yeah, that's good.
Hi. That's a lovely talk. Enjoyed it a lot. Wondering if in the AMR detection studies that you did, there are all these theories that talk of cycling of the antibiotics versus using them simultaneously in the hospital settings that alter the prevalence of these AMR strains.
Right.
Is there any correlation you found with the usage of the antibiotics and the prevalence of these strains?
Yeah, so this is one of the questions I'll reformulate that. So what in the initial study about recovery that I described, this is a mishmash of studies from around the world, and each study had its own. Many of these studies were controlled studies where they had, say, coamaxiclov as the antibiotic that they gave to healthy people, and they studied. There are some other studies which had cephalosporins. We had a range of antibiotics. So we did not, in this study, because of the size of the cohorts and so on, we did not pay attention to which antibiotic was given. We were trying to find general phenomena. But in follow-up studies that we're doing now, we're trying to control which antibiotics, and we're trying to do studies with multiple types of antibiotics, clinically useful, not the toy studies and then try to ask this question, is there differences in the key bacteria that are important for recovery? But the question you asked, sorry, it's a different one. It's an even harder one, which is the cycling of antibiotics and so on. I think those kind of studies are very hard to do. I think ethics for taking healthy individuals and giving them cycling of antibiotics is going to be, and clinical situations are always really determined by the pathogen, the infection that the person has and what is needed. So you can't control that too much either. It's challenging to do the studies. Yeah.
Okay, thank you, sir.
No.
So, it's done. So the next speaker is Dr. Shruti.
You can go.
Okay, okay.
I'll go.
Can I change my PDF? I have a flash talk at the last session. This is
No,
my
no, no. Both the sessions will happen now simultaneously. You want to change your PDF? Yeah, you can upload your file. You have it send or email it. Yeah, and if there's an IC ID over there also.
My class is available at Rajendra Prasad sir.
Yeah. So first flash talk will be presented by Mohammed Shabir.
Am I audible? Yeah, hi, I Shabbir Ahmad working with Sunil Laxman at DBT in STEM. First, I would like to thank the organizing committee, the IBSE, given me the opportunity to share my work. The title of my work is Pool of Amino Acid Function as a Public-Private like resource. What I thought over 3 days we enjoyed a lot of discussion, poster discussion and metagenome and microbiome diversity. But we don't know how microbiome existing together and how exchanging the metabolite. Coming to the brief introduction of my topic, nutrient exchange is widespread phenomena occurring in nature, but we don't know how much metabolite produced by the cell and which metabolite secreted by the cell. And majority of the microorganisms existing in nature are highly complex community and exchanging these metabolite happening in nature. Among these metabolite exchange, amino acid dedicatedly exchange among the microbial community. Majority of the microbial organisms are auxotrophs in nature. The amino acid exchange is widespread phenomena happening in nature. And then we are asking the question we don't know which cell producing which amino acid and which cell consuming which amino acid. Based on this thing, we are asking the question amino acid production, consumption, secretion at a particular time point. And we are asking the question how much amino acid produced by the cell and how much amino acid secreted by the cell over a period of time while cells are growing under minimal media. Here we have taken each stage a model system and asking the question how much amino acid production, secretion, consumption in various phases of growth while Saccharomyces cerevisiae growing under minimal media without amino acid, glucose as a carbon source, ammonium sulfate as a nitrogen source. Here the simple illustrated suggests the growth as well as nutrient consumption, and we measure the rate of amino acid production in various phases of growth. From this data suggests the majority of the amino acid production is higher at earlier phases of growth, but over period of time amino acid production decreased. And this suggests the summary of this amino acid production. From this data, I haven't suggested the amino acid production consumption together, but we know the rate of amino acid production. Then we are asking the question, what are the total amino acid at a particular cell? When we went for the intracellular amino acid at various phases of growth, and we measure the absolute quantification of intracellular amino acid at various phases of growth, we found the amino acids are dynamics, not a similar over all the time. It suggests the glutamate and glutamine is higher over period of time, and some amino acids are least at earlier phases of growth, but over period of time increased. And these are suggest the clear distinct intracellular amino acid in various phases of growth. While we measure the extracellular amino acid, what we found the extracellular amino acid are not statics while the cell secreted at particular time point, but over period of time some amino acid are decreased and some amino acid are increased. It suggests that might be cell while they secreted the amino acid, again cell retaking back. Then we try to find out what are the correlation between intracellular extracellular over period of time, and we classify the amino acid based on the extracellular and intracellular, and those amino acids secreted by the cell, it come under public good because those amino acids can be consumed by any group of cell within this population, and some amino acids are not secreted by the cell, but over a period of time might be low or might be high inside the cell. Those come under privatized source. And these fall under public resource amino acid these, public goods amino acid these, and private goods these. Then we make the engineer from this group public good auxotropes and private good auxotropes, and finally try to make the synthetic community how the synthetic community is working. Here these two auxotropes, public good auxotrope and private good auxotropes, those auxotropes unable to grow under minimal media, but while both public good auxotrope and public good auxotrope combining together and growing healthy, here the overall data suggest the population is maintaining over period of time. But while public and private auxotrope growing together, the public auxotrope growing healthy, but private auxotrope can't able to grow. This suggests that each stand complementary, not able to support each other. Finally, we propose the pathway. We propose the model amino acid production. Some amino acids are privatized by the cell, and some amino acids are secreted, and those amino acids secreted, they supported the growth of other community, and some amino acid is again reuptake by the cell. Finally, we propose the model a synthetic yeast community of stable yeast community. And finally, this is the acknowledgement. I highly acknowledge the Bliss community and serve for funding, and these are the lab members, and highly acknowledge our supervisor, Dr. Sunil Lakshman at DBT in STEM. Thank you.
Thank you, Shabir. Any questions? No. Thank you. We'll have the next flash talk from Pratyay Sengupta.
Hello. Yeah. Just check. Hi. Yes, I'm audible. So, hello, I'm Pratyay Sengupta, and I'm going to talk about microbial interactions in built environments. So what is a built environment? Built environment is essentially what is built for human, what we see around us. And it is important to understand microbial interactions in built environment because built environments are a reservoir for a lot of unique microbial communities, and they are associated with a lot of human-associated microbiome. They are barriers for exposure to natural environments, and there are beneficial microbiome. And then they reduce microbial diversity and can accelerate a lot of chronic diseases. Not only that, built environments has a lot of effect on establishing gut microbiome in the children. And so in this study we are trying to get the built environment microbiome from different environments. Like we have samples from hospital environment as well as metasub, which is we have multiple speakers discuss that, and then we have from office environment. And we are trying to address three simple questions. Number one, what is the microbial ecology of these built environments as we spend a lot of time in these environments? Are there any built environment that play key role in the microbial interactions? And then what kind of cross-feeding that are shaping this kind of environment? So we observed that from these three environments, there are microbial diversity. They are distinct microbiome. They are very different from each other. And if you notice in the class level diversity, we have seen a lot of bacilli, clostridia, gram proteobacter, which are very abundant in hospital environment, but not in metasub, which is metro station, as well as in office, which resembles our home. And they are known to harbor a lot of nosocomial pathogen in these classes. So that is again what we observed in the hospital environment. So we see that these environments are very different. We try to address what sort of microbial interactions are there. We constructed microbial interactions network, co-occurrence network, where each node is a microbe, and the age between them is a probability of they being co-occurring in that particular environment. And as you see that in the node-wise, the bacterial features are very similar. However, the interactions are very different between in these environments. So we segregate these ages to see what are they. So we had one generalized age which is present across multiple environment, and then if it is present in only single environment, we call it specialized age, which is again segregated based on whether the nodes or the bacteria are seen in different environment. But we observed that the fourth thing that I will ask you to note that is the specialized age, where the nodes which are known to be present in other environments. So that is with both generalized node, and that covers a lot of like part in the hospital as well as metasub and a bit in the office. So that shows that these particular microbes are present in multiple environments, but interacting particularly in this environment or particularly to these environments, right? So that is nearly 30%. So like we then try to ask what kind of interactions are there. So for that, we from the network we get a subset of this thing. We define different communities of different sizes, and then we iterate them with to calculate two scores. One is cooperation, one is competition. They are called metabolic interaction potential and metabolic resource overlap, to identify what sort of interactions they are having across this environment. And I'll just quickly show you one of the results where we have seen, we plotted MIP with MRO, where in the MIP, if you go more right side, that is more cooperative, where if you go in the y-axis top, that is more competitive. And as we increase community size, we see a clear distinction in the groups. And as you can see, office is more competitive environment, whereas hospital is more cooperative, metasub being in the middle, and random is all over the places. So this actually probably tell us that metasub, being a public transport domain, could help to disseminate these different microbiomes from different resources, and that's why we are seeing these communities in the middle. So I would like to summarize what I said. Just so it's a systematic approach of combining both network biology as well as biological component from the metabolic modeling using nearly 600 shotgun samples, and we observed that microbiome across these built environments are very diverse and distinct. We have seen a lot of nosocomial pathogen present in the hospital environments, and then they could be potentially associated with healthcare-associated infections. And then this is very important to understand microbial interactions using different computational approaches. And we could extend this to possibly disrupt these communities with another pathogen or different microbes from different environments to see how this community behaves. So it's important to have a surveillance in these environments. With that, I would like to thank my lab and welcome again to this campus. I hope you had a nice 3 days. Yeah.
Thank you, Prate. Any question?
Yeah.
Thank you. Great talk. Just a curiosity question. Have you compared with other built environments, like, for example, rural homes, for example? There's public data available. Might add more value to see what kind of, diversity exists in rural homes compared to urban homes.
Yes, not at the home scale. We would like to do, but we chose these 3 datasets because they are from the same environment, and yes, that is the main idea of having these datasets.
Thank you, Prate. Yes. Our next flash talk is by Mohan Maruti.
Good afternoon, everyone. I'm Mohan Maruti. I work as a research associate at Cancer Institute. It's just an adjacent campus to IIT Madras with Dr. Mailwahan Bose. So, colorectal cancer is the second leading cause of deaths among all the cancer-related deaths globally, and is the 7th most common cancer in India. According to the incidence rates, it was high in the developed nations, but now in the developing nations also it is picking up. So recent research studies show that there is a gut microbiome dysbiosis that leads to the CRC, and they have identified some of the oncomicrobes such as PKS and then enterogenic Bacteroides fragilis and then E. coli that produce toxins which will lead to the induced genetic mutations and alter the host gene expression. So actually, most of the studies were on the developed countries. A recent study shows that of approximately 4 lakh microbial samples collected across 2,000 odd studies. So there was a representation of greater than 70% from North America and then Europe. And then South Asian countries contribute only to 0.18%. So to overcome this imbalance, actually there was an international CRC microbiome was established, and then to look at the microbiome, advanced microbiome studies across the developing countries like India, Vietnam, Argentina, and then Chile. Our main objectives in this study is that we want to understand how the gut microbiome composition changes across the CRC patients when you compare to other countries, and then across Indian CRC patients, and then how does it differ from the healthy individuals. Based on this, we want to actually construct a machine learning model that can actually distinguish between CRC and then non-CRC patients. So these are the data sets that we have collected. So this data set actually consists of like 46 CRC patients and then 43 healthy patients. So this we have collected at Cancer Institute, Adyar, Chennai. And then before this, there was only one study, in Indian study, that consists of Gupta et al. They have collected it 30 CRC patients and 30 healthy patients. They collected it from Bhopal and then Kerala. And then the other data sets were actually collected at these respective hospitals. So this is our protocol. So first we will collect the fecal sample of patients and healthy individuals. Then from the GFOBT card we will extract the DNA, and then we do the 16S amplicon sequencing, and then we do the microbiome analysis using KIME, and then we input that data to the machine learning model. So what we observed actually was like when we did this analysis, so across different countries, so it was separated when in the PCOA analysis, it was separated across the geographies, but not with respect to the cancer status. So the South American, and then Kyle presented the left side of the plot, and whereas the Vietnam is present at the top, and then India at the bottom. So it's more of related to geography than related to the cancer status. Then we have looked into the taxonomic abundance. We thought like among the Asian cohorts, the Privotella was major, and whereas in the South American counterparts, so the bacteroids were actually the major contributors. And among all the cancer data sets, so the Escherichia shigella was actually predominant. Then we looked into the earlier study. We compared our study with Gupta et al. Study. So there also we could see that Privotella is major among the Indian data set. And then here, one thing we could not understand is that in CRC, the bacteroids were major in Gupta studies, whereas in our study, in healthy controls, bacteroids were more. We could not understand that. So then we did a diversity analysis, and then we found that South American samples are highly diverse compared to India and then Vietnam. And then we compared our study with Gupta et al. Study. So our samples are more diverse compared to Gupta et al. So we could not understand whether it is due to this geographical or due to any technical thing, because they have done shotgun metagenomics and metagenomics for the data, and then we done 16S amplicon sequencing. So we do not clearly know this thing. So next we have built in machine learning model using random forest classifier. So in the current data set, we got an AUC of 0.86, and what we have done is that we have taken a data set that is published by Young et al. Using UK data set, and then we tested our model. In that model also our model could predict with an AUC of 0.86. Next, actually, this was the work done by a 11th class student, as morning Anshu madam was telling. So she was selected for IRIS, and then she is comparing Indian and then Sri Lankan samples. Here also we could see that Privotella is major, but we need to look into much more closely. And then what we are doing as a next steps is that because there are not enough samples, we thought we will actually collect samples from various parts of India. So now we have only representative samples in around Chennai, but we have partnered with other institutes and then hospitals. So they represent northwest, east, south, and then we will collect all the samples. And the target is like per institute, if we collect like 350 CRC and then 350 LT samples, and then if we do this kind of analysis, we will get an idea about how microbiome actually changes in CRC and then healthy patients across the India. So these are conclusions. As I said that Escherichia shigella is more common in CRC, and then we observe larger amount of microbial diversity in CRC patients, and there is a geographical separation that is observed rather than by status. So I would like to thank funding agencies, International CRC Microbiome Network, and then DBT Wellcome Trust India, and then Peacock Project.
Thank you,
so
Mohan.
much.
Any questions? No questions? Thank you, Mohan, again. We'll have Prashant Dhumpuri next for the flash talk.
Yeah, it's on now. Awesome, thank you. Can you hear me? Hey, so I'm Prashant Dhumpuri from Microbiotics, a startup company based in Bangalore. If you're in this room, you probably heard that gut microbiome is related to our health. What does not get talked about a lot is how messy the sample collection process is, how expensive genomic sequencing is, especially in economically challenged countries like India, right? And the report generation for genomic sequencing takes between 4 to 6 weeks. So we at Microbiotics are addressing those. We want to bring gut microbiome testing into clinical adoption, and these are the issues we've addressed. The work presented here today is an extension of the work that we've already patented and commercialized. We have conducted clinical trials throughout India, where in trial participants, we collected finger prick-based blood samples. We measured metabolites from those samples using untargeted mass spectrometry. For the same trial participant, we collected their stool samples, did genomic sequencing, and measured their microbes and abundances. So for a given participant, we have both metabolites and microbes. We fed this to a neural net model. In particular, in this work, we fed it to an encoder-decoder type network. So one network that can predict microbe and their abundances, and the same network can predict disease labels and disorders. So think about this, right? So now, using the predictive power of AI, if I can collect just your finger prick-based blood samples, I can predict microbe and abundances, and I can predict your disease state or disorder labels. So no more messing around with stool samples. This particular work, we validated this at one of the premier hospitals in India, a clinical trial of 93 patients, 3 cohorts, healthy controls, obesity, type 2 diabetes, so that's T to D. We collected both finger prick blood tests and stool samples. So we are still saying stool samples is the gold standard. So NGS detection, that column is your stool sample microbial abundances measured using stool sample. If the gold standard detects a microbe and our model predicts the same microbe within the 95% confidence interval limit, then we're saying it's a true positive. And so on, right? So if your NGS does not detect a microbe, but we predict it to a 95% confidence interval, we're saying it's a false positive. So using all these metrics, we computed accuracy, sensitivity, and specificity, one network without stool samples, with just finger prick-based blood samples. We are 95% as accurate as a stool sample-based method. Since this was a network that could also predict disease labels, we also predicted disease labels, and we are touching 90% accuracy. And where we erred was in the obesity and type 2 diabetes cohort, two metabolic disorders that are often linked to each other. So these results are actually very encouraging, and we want to extend our already published work into diagnostic space. So in a sense, this is what we do at the company. Finger prick-based blood tests, no more stool samples, cost-effective. We turn around your reports about 75% faster than genomic sequencing, and we're hoping that this is going to lead to improved adoption. Questions?
Thank you, Prashant. I have a question. So, here.
Oh, thank you. Sorry.
I have a question. So, what do you feed into your model apart from just the presence? I mean, the microbial species name. I mean, do you have some biology that connects the microbe and microbial metabolism?
Right. So we feed into the model microbes, the abundances of microbes at the genus level. Right now, we are doing it just at the genus level.
Okay.
And the relative abundances of metabolites. So metabolites are the biomarkers that microbes release into the bloodstream.
How do you distinguish between the microbial versus like
Right.
host?
So we have a data set that we've collected where we took stool samples from participants, and then we did metabolomic analysis of the stool samples. We got those metabolites. And there are databases like MetOrigin that gives you the origin of each metabolite. So combining those two, we know what the origin of a metabolite is.
Okay. Thank you. We'll move on to our next flash talk, which is by Amit Yadav.
Hello. Okay. Good afternoon, everyone. First, I would like to thank the organizers for providing me with this opportunity. I'm Amit Yadav, an ACSAI PhD student at CSIR Institute of Microbial Technology, Chandigarh, supervised by Dr. Sri Krishna Subramanian. I'll be talking about the approach for improved biosynthetic gene cluster detection in PacBio Hi-Fi read metagenomes, Hi-Fi BGC. Biosynthetic gene clusters are the set of genes that are physically co-located on a genome and encode for secondary metabolites that have applications in medicine and agriculture. BGC prediction in metagenome enables exploration of biosynthetic potential in an environment and allows access to novel natural products. BGC prediction in metagenomes typically involves two steps for both short read and long reads. These are first taking the input reads and assembling them using an assembler, and then on the obtained context, run a BGC prediction tool such as anti-smash. With short reads, we are more likely to get partial BGCs. With long reads, we are more likely to get complete BGCs with all the genes intact. With this introduction, our goal is to predict BGCs in PacBio Hi-Fi metagenomic data. The typical approach for doing this is we start with the initial raw reads and then assemble them using one of the three Hi-Fi tailored metagenome assemblers, Hi-Fi SM Meta, MetaFly, or HiCanu. Then on the obtained context, BGC prediction is performed using a tool such as anti-smash. These obtained BGCs are then clustered using Bigscape clustering method, and then these are being reported. In our proposed approach, we start with the initial Hi-Fi metagenomic reads, and then assemble using all the three assemblers, Hi-Fi SM Meta, MetaFly, HiCanu. The context from these three assemblers are then concatenated together into a single file. Then we take the initial raw reads and map these reads to the concatenated file using Minimap2. Then from this, we obtain the unmapped reads using SAM tools. Now we have 4 sets of sequences, the 3 assemblies, and the unmapped reads. On these 4 sets of sequences, we run anti-smash for BGC prediction, and then take all the predicted BGCs together and perform a clustering using Bigscape. For each Bigscape cluster, we assign a representative BGC, and then these are being reported. Here we compare our method with the existing approach on 4 data sets. Here I'm showing 2 of them. On the left figure, on the x-axis are the methods, and y represents the count of representative BGCs written by each of the method. Considering the typical approach, we define 3 different methods based on the assembler used in the first step for assembling the reads. These are the first 3 methods on the x-axis, and the 4th one is our proposed method. And we can see the Hi-Fi BGC returns the maximum count of representative BGCs. BGCs are being further divided based on whether they are partial or complete. And the same observation can be made for the human data set as well. Here, the abstract plots demonstrate the contribution of the individual assemblies and the unmapped reads to the final output of Hi-Fi BGC. Focusing on the upset plot on the left, the second bar from the left shows the count of Bigscape clusters that contain at least 1 BGC from unmapped reads. This third bar shows the count of the Bigscape clusters that contain BGCs exclusively from Hi-Fi Meta. Subsequently, the subsequent bar from the Metafly assemblies, and then there is another bar showing BGCs exclusively from Hi-Canoe. This clearly demonstrates the benefit of using multiple assemblies and the unmapped reads, and the same observations can be made from the human data set as well. Here are 2 select Bigscape cluster visualization examples. The first one on the top shows 3 BGCs from 3 different assemblies, Hi-Fi Meta, Hi-Canoe, Metafly. The arrows represent genes, and the vertical lines denote the sequence identity between them. Here, the 3 BGCs are 100% identical to each other, and represents a good case where the same BGC is being identified in all the 3 assemblies. Whereas in the figure down below, we have a partial BGC for Hi-Canoe assembly, and this demonstrates the benefit of using multiple assemblies, as now this portion, this missing portion of Hi-Canoe BGC would be recovered by the other 2 assemblies. Hi-Fi BGC is provided as a command line based tool on GitHub and is implemented using Python and workflow manager SnakeMake. This work has been published. I would like to thank my supervisor, DBT Bink, and Bioinformatics Center IMTech for this. Thank you very much for listening.
Check. Thanks for the nice presentation. So I've got a couple of questions. How did the Hi-Fi BGC fare in environmental samples? Like, were there any significant improvement in predicting the clusters from the environmental metagenome samples?
So one of the samples we tested was for sludge data set.
Sludge, okay.
Yeah, so it's performing similar across the 4 data sets we tested.
Okay. And when you say about Hi-Fi, you're very sure about the quality scores of the Hi-Fi reads?
Yeah, these are really good, highly accurate, 99%.
Yeah. How does the platform fare for the other data sets from Nanopore or
No, this is specific for Hi-Fi.
This is only for the Hi-Fi?
Yeah, because I'm using Hi-Fi tailored assemblers, specific assemblers. So this is specific for Hi-Fi.
Okay, thank you.
I have one question. You have shown the gene predictions using your dataset. How it is comparable with other gene prediction tools, like which is available, like Prodigal and Proca? Have you compared that one?
Okay, so I'm using a method anti-smash, and it underneath uses Prodigal for gene prediction.
Okay.
So the way anti-smash works is it predicts genes using Prodigal, and then finds out certain domains which represent BGCs. Okay. So this is the way anti-smash works.
Okay. And also another question that he has also asked, whether this data, whether you, because in one step you saw that you have used 3 different assembly tools.
Yeah, right.
And now you merge them in one. Can we apply this for the shotgun Illumina reads also?
This can be looked into, but I have not looked into it. But I doubt there will be much benefits. This is my.
No, I think it will be good because there are 2-3 different type of assembler is available for the shotgun genes. like what is called the mega heat or spreads. But if we do all type of, we get different number of contexts, and if we can merge these two, like what we do for the math preparations, where we can make the max using 2-3 different tools and we merge them. So I think it would be beneficial for.
Yeah, it could definitely be looked into. Yeah, right.
Thank you.
Thank you, Amit. Our next flash talk is by Abhishek Gupta.
Hello. Oh, thank you everyone, like inviting me and like giving a flash talk option. Like, so I'm Abhishek Gupta, working in CELSI NUS. So I'll be working on like how the iron mediates the methanogenesis. It's just quite different topic because everyone is covering gut microbiome. Now we are discussing how this greenhouse gas is basically generated or is stimulated or inhibited by the iron mediated metabolism. So you know, like we are living in the industrial era, which has more than 2.21% of methane in the atmosphere. So do you see like methane, N2O, and the CO2 are the major greenhouse gases, and they have the methane has more potential of methane of the climate change in Earth, which is 28 times more potent. Why it is called, why it has more potential of greenhouse gas or the climate change? Because it captured 28 times more the heat as compared to CO2, and N2O captured 265 times more heat as compared to CO2. If we want to like regulate the climate change, we have to first like limit the production of CH4, because the lifespan of CH4 is basically our decade, 10 to 12 years. But while for CO2 and N2O is around like hundreds to thousands years. So if you want to see like immediate effect in the next 10 years, you have to reduce the amount of CH4 production immediately. So from where this methane is coming, the most of the methane is coming from the methanogenesis events. Okay, so nearly like 50% of the global methane emission is coming from the various aquatic ecosystem, including wetlands, freshwater estuaries, the coastal area, aquaculture, and the marine ecosystems. Similarly, there's another study which reported that global methane emission is coming from the major riverine streams across the globes, and you can see the Southeast Asian countries, this area like the Africa and the American continent is basically has more methane, as well as they emit more methane from the riverine systems. But there is a term called urban waterways. These waterways are basically a channels, or we can say a network of reservoirs, as well as the stream, ditches, and canals, which basically helps to regulating the storm water harvesting systems. Basically, Singapore is, even though a very small continent, but it has a very huge network of urban waterways like the Amsterdam. So these waterways is basically a system of reservoir. You can see a very bright, the blue color, a big dot. It's basically a reservoirs, and the lines basically are various streams, which covers around 7,000 kilometers in Singapore. So right now, these are major hotspot of the methane emission, as we have seen in the Western countries also, like in Amsterdam and some other European countries, which shows that there's a methane cycling microorganism, as well as ditches and canals can emit more methane than the freshwater lake system. So these are understudied environment, and we want to see how methane cycling is happening in the riverine system of urban waterways. So since in my title, you have read that we are working on the role of iron methanogenesis. So you know iron is the fourth most abundant element in the earth crust, and it has a more redox state, like it can goes to the Fe2+, or it can goes to the Fe3+ based on oxidation and reduction potential. So there are different kind of iron minerals like ferrihydrite, goethite, hematite, magnetite, and these minerals are basically either amorphous in nature or they're crystalline in nature. So based on this crystallinity and the amorphous nature, they can help in the transfer of electron from bacteria to the archaea, because archaea needs electron to convert CO2 to the methane. So if they promote, it means they are more crystalline in nature. Sometimes they inhibit also, because of iron reducers have basically they outcompete the growth of methanogens, or sometimes they are toxic to the methanogen also. So these are the different mechanisms through which like methanogens takes electron from the bacteria, they can directly attach to the archaea, through their pili or nanowires, or they can, methanogens can obtain like electrons either from the metabolites that are produced by the bacteria like hydrogen, formate, etc. And they can directly use cytochromes of bacteria for the transfer of electron to the methanogens. And the last one, which I am dealing with, is basically the conductive materials. So like this, if this is iron minerals, so bacteria will touch, like basically attached here and archaea attached here. So there's a direct electron transfer from the bacteria to the archaea with the help of these conductive minerals. Can be iron oxides, can be manganese oxides, or can be any clay or any other minerals. So with this, the aim of our study to understand the role of iron on methanogenesis in urban water, freshwater sediment using microcosm-based study. So to understand this, we have designed 10 different setup, basically the experimental conditions. So we collected samples from this urban canals, which is basically a Kalang River, which is close to the Angmeo, it's close to the park area. And then we collected the samples, we put in different bottles, and then we added 3 different types of organic carbon. One is acetate, propionate, and butyrate, and there's a mix of all these 3. Why we have chosen this? Because these are basically the intermediates of organic matter decomposition. And then we added iron with these organic carbon. So we have a 4 set of organic carbon, and we have a 4 set of organic carbon with iron. And we have one unamended setup, where we have not added anything, only water in the sediment. And we have added only iron as a iron amended control. And we measured, we kept it for 10 weeks, and every second weeks we did the sacrificial type of sampling, and we measured all these parameters, including the gas data, amplicon, and we performed the shotgun metagenomics. So let's see how the gases are generated in these type of setups. So you can see it's not that very clear, but the bold line is basically where we added iron, and the dotted line is basically where we have not added iron. It's only the organic carbon amended setups. So what we have found that organic carbon amended setups had showed higher amount of methane generation as compared to the iron plus OC amended setups. While in case of carbon dioxide, it is basically vice versa. We have observed like maximum CO2 production was observed in case of iron plus organic carbon amended as compared to only organic carbon amended. It means the syntrophic or the fermentative populations which are utilizing this acetate, propionate, or butyrate, they produce CO2 or the acetate, but because of iron, there is a reduction in the methanogenesis. So now we are interested what are the microbes which are involved in this enrichment setups and helping the promotion of like generation of methane or the CO2. So these are the major 4 microbial archaeal taxa, which are basically methanogens. And what we observed, the Methanosarcina, this one, is basically all this color represent the different setups. So what we have observed, these 4 bars you can see, it's quite higher in abundance. So they are all enriched in organic carbon plus iron amended setup, while rest are like we can see, if you see the counterpart of this to this, so they are enriched only in organic carbon amended setup. Same for the syntrophs. What we have observed, they are basically substrate specific. Some syntrophic member require propionate, while some other require the butyrate, or some are like generalist, they are growing in all the propionate, acid, and butyrate. So we also recovered nearly 300 max from these data sets, and what we observed, there are different, all these max at least contain a single type of iron reducing genes, because there are multi-heme cytochrome type of iron reducing genes, there are flavin type cytochrome iron reducing gene, and there's a homolog of multi-heme cytochrome. So we observed like at least one of these genes are present in this max, and they also have a C1 metabolism genes also. So now we are trying to understand to see their functional notation of how they are degrading butyrate, propionate, acetate within the MAG. And with this, I would like to conclude our study. So we added propionate, acetate, and butyrate in our systems. And what happened, it helped in the enrichment of the syntrophic populations, which later on produce acetate or CO2 and hydrogen, and which help in the enrichment of acetoclastic or hydrogenotrophic methanogens, which produce methane. But what happened when we added iron along with this organic carbon? What happened? The syntrophic population was basically doing their work, but iron basically reduced the production of methane. It may be due to substrate competition between the iron reducers as well as the methanogens, or it might be the iron that we added is toxic to the methanogens, and we have seen that methanosarcina, which are enriched only in the iron amended setups, is switch their role from the methanogenesis to the iron reduction, because there are certain species of methanol sarcina have the capability to do iron reduction too. So now we are doing another study where we are using different iron oxides like hematite, goethite, magnetite, and ferrihydride to check how they are behaving in methanogenesis event. So with this, I would like to acknowledge my boss, Sanjay Swarup, my team member, Andrew is here also, and other members, Celsi, my office, and the PUB, who basically help us with sampling these sites. Thank you. Happy to take any questions.
Thank you for the enlightening talk. I had a question that iron cycle, methane cycle, and sulfur cycle, they are kind of correlated. So when you are checking that iron is affecting methane, like it's reducing the methane emission rate, is it that somehow sulfate reducers are activated in the process? Because sulfate reducers, they are like really notorious ones. They go hand in hand with methanogens. So maybe in a condition where acetate or CO2 is readily available, it's a very good opportunity for them to grow. So it may be so that it is not actually the iron which is inhibiting methane emission, but it is actually the sulfate reducers which are doing this. So did you check on the role of sulfate reducers?
Yeah, I have checked in our data. We haven't found any sulfate reducer whose abundance got increased substantially in our data sets. So even there are some sulfate reducers have the potential to reduce iron also. So like desulfitobacterium, so they can do like dual role, sulfate and iron also. But in our case, the amount of sulfate in the sediment is quite low. So the chances is very less. So while collecting these samples, we have done some experiment to check what is the initial amount of the GSGs there. So it's quite low, first of all. And second, like you can say, like we collected the samples from the field. We immediately basically sealed in the bottle and kept it there for a long time, at least 6 or 7 months. And after 6 or 7 months, what we observed that the color of the sediment changed to black. It means sulfate reduction as well as iron reduction are going simultaneously, and we can see a black precipitate. But in this case, since we added iron, the chances of getting enrichment of sulfate is quite less. Even the amount of sulfate is quite low in our sediment sample as compared to the iron, because total iron in the acidified fraction is close to 12,000 mg per kg, while the soluble fraction is close to around 400 microgram something. So in our case, no sulfate reducer is not enriched.
Thank you, Abhishek. Our next flash talk is by Rajendra Prasad Sahu.
Hi, I am Rajendra from IIT Kharagpur. Before going into my topic, I would like to ask you one question. Do you know a significant fraction of the microbial community is residing within the earth crust? So, and our knowledge regarding them is very less. So, scientific deep drilling done by the Ministry of Earth Science at Koina seismogenic zone of the Deccan Trough provided us a unique opportunity to investigate such type of microbial lives which are residing within the Archean granitic basement of the Deccan Trough. Here the rock cores recovered from these depths were analyzed using the geomicrobiologically to investigate the inhabiting microbial communities, their geochemical drivers and all. So overall geochemical analysis suggested that this environment is a progressively hot environment as well as the oligotrophic environment. If at all microbial life is surviving over there, they have to withstand the extremities which are prevailing over there, and as well as they have to follow the chemolithotrophy. So let's see if any organism is present over there. The cysteinase RNA gene amplicon sequencing suggested that the presence of highly diverse microbial community and a very few number of OTUs with high individual abundance, they were found to present all over the 7 different depths, which suggests that their superior metabolic ability and survivability. However, a large number of OTUs, around 40,000 OTUs with less individual abundance, they are sample specific or depth specific, which suggests that there is a significant role of the local environment on the community. So, to investigate the role of local environmental communities or different type of stochastic and deterministic factors on the microbial community, we did the phylogenetic distance-based null model analysis
And it suggested that there is a significant role of the variable environmental conditions on the microbial community. It is suggested that if there is a significant role, if a community is experiencing this, the variable selection, so the community will be less interconnected. So to check the community interconnectedness pattern, we did the question analysis and we got the same result. We can see that the community is less interconnected. Now, to check the community interaction or community behavior, we did the co-occurrence-based network analysis. We found that there is a predominance of the positive interaction in all the cases than the negative interaction. We suggested that the community members are involved in mutualistic interactions in this environment, which might due to utilize the available nutrients efficiently. And when you see the pattern of the interaction, with depth, we see a distinct pattern with reduced in community interconnectedness and increase in positive interaction with depth. We suggested that there is something has happened. That means something alteration has been happened in this environment. To find out the alteration, if anything has happened in this environment, we try to see the borehole log data. And this I have here I am showing this data where we can see that they have during drilling the borehole, the Ministry of Earth Science in association with ICDP, they have measured the borehole logs and they found the presence of the fracture zones after the 2100 meter accusometer, which is following this region. We suggested that there is some relationship between the seismic activity which has created the fracture over there with the microbial community interconnectedness pattern of the microbial community. To check this, what we have done, we have tried to compare the community interconnectedness pattern with the helium ion concentration. Why helium? Helium is an inert gas, and after its production from the magma or the radioactive decay, it gets accumulated into the fracture zone. If we are getting a higher fracture zone, then there will be a higher amount of helium we can expect. So when we compare the community interconnectedness pattern with the helium ion concentrations, we found there is a strong correlation between them. We suggest that the community is experienced more strongly by the seismic activity which is happening over there. So in conclusion, we can say that in this oligotrophic environment, the inhabiting microbial community is shaped by the local environmental variabilities, and the microorganisms are involved in a mutualistic interaction to utilize the available nutrients more efficiently, as well as withstand the prevailing extreme conditions. And in future, I would like to investigate other life forms like viruses, fungi, and eukaryotes which are inhabiting over there, and also elucidate the influence of the inhabiting community on the geochemistry and the lithology of this environment. With this note, I would like to thank Dr. Sukant Roy from the BGRL Minister of Earth Science, Dr. Thomas Weisberg, ICDP, and IIT Kharagpur, as well as the CSR for funding me for my PhD fellowship, as well as the organizer of this conference, Karthik Raman sir and Karthik Anandaman sir, as well as the Kasturi sir, for providing me the opportunity to present this work here. Thank you.
Thank you, Rajendra. Our last flash talk for today is going to be presented by Preetam Kundu.
Hello, am I audible? Hello. So, good afternoon everyone. Thanks for the opportunity. So, today I'm going to talk about the story that I have covered during my PhD. So, just completed in this early 2024. So, from IIT Kharagpur. Right now I'm a postdoc at IBSE, Chennai. So, this was the story that I covered during my PhD. Like I started with some metagenomic data analysis and tried to construct a global metabolic interaction network just to understand like what are the metabolic crosstalk that is happening inside a microbiota. Specifically, I work with termite or insect gut microbiota. So then with this global network analysis, I have identified some keystone species, the story that I'll not go into detail today. So then I moved to another method that's called genome scale community modeling. So I think you got an introduction of this method already in this conference. So here I have tried to map these keystone species that I have identified from this metagenomic analysis and looked into in depth into their biochemical pathways to understand actually what are the metabolic exchange or crosstalk that is happening within the community, so that the community can survive or can have a higher metabolic activity than the individual microbe on their own. So that was the overall idea behind the story that I covered during my PhD. And at the end, the story that will be that I will be telling today for 2-3 minutes, coming to 3 minutes. So that is how I can use this model assistant platform to track the microbial interaction in a smaller scale, so that I can have a synergistic microbial community through an integrated modeling and experimental approach. So the approach started with that. Like I have isolated some 6 bacterial species. Those are the lignocellulose degraders. As I am enriching this microbe in lignocellulose media from isolating from the termite gut. Termite is the base wood feeders or the microbial community residing in the termite gut are one of the major lignocellulose degraders that has been identified till now. So now what I am doing here is I'll go into the detail. Then I using the FBA and the FBA like the flux balance analysis tool and trying to identify what would be the growth and metabolic synergism within these microbial species that I can predict from in silico, and can I check that with some experimental assay at the end towards the end. So what I have started with the isolates and checking them in different substrates. So as you can see, there is one microbe P1. I'm calling it as P1. So it is not giving me a zone of clearance in case of Xylan, although it is growing. But in case of CMC, it is giving me a clear zone. But in case of the P2, you can see it is not giving. It is giving me a clear zone is Xylan, but not in case of the CMC. So then I have done a 16 years to identify what could be the species and identify them, and looked into their whole genome sequences from the NCBI. Then I also performed the enzyme assay. So not just these qualitative matters to check how they are active in different substrates. Like this is the FPS activity, as you can see for the different microbes that I have identified. This is the CMCS, that means the endoglucanase. This is the total cellulase, and this is the xylanase activity. So to summarize it, that what I saw is that the microbe performing in one substrate well, it's not necessarily performing good in other substrate. So I have checked like whether actually there is a substrate specific limitation for each of these microbes, the 3 substrates that I'm using here. So now the idea was that as I already during my PhD I have worked into this community genome scale community modeling. So I think like if I can combine these microbes, and if this substrate specific limitation of this individual microbe can be overcome, so I have looked into I have constructed the possible, so we can have a 15 possible microbial pair if we are pairing it in a pair of 2, and then I have used this PMA and the PGSI analysis, a similar type of method. I think Karthik Raman sir have already discussed with you in this meeting. So the idea is that, so I don't want to have tackle all these 15 possible pair, because I have already 3 substrate that I need to check. So it will be a huge task as a experimentalist. So I'm just reducing the number like that. My model are giving the prediction which could be the possible synergistic microbial pair. Those will act well in a experimental design. So now with this PGSI, what I'm trying to see, like which are the microbes those can give me, these are the prediction that I'm making, that those can give me a positive or mutualistic interaction while I'm culturing them. And then coming to the PMA, these are based on the flux variability and the reaction that has been opened up. That means these are the, so I am calculating this parameter based on how much reaction has been opened up or how much flux space has been expanded when the microbes are in the community, not compared to its individual model. So now by analyzing these parameters, not going into the detail, so I have identified some 9 microbial pair or some crucial microbial pair that I have experimentally experimentally tested with the same 3 substrate, that you can see, and in some of the cases I got really high DES, that I'm defining it as a degree of enzymatic synergism. That means they are acting well in community compared to their individual while they are growing individually. So these are the some base microbial pairs that I have got, but I haven't got any microbial pair. Those are actually performing very well in all of the 3 substrates till now. So then I looked into the same method and going into a combination of the 3 microbial community, you can understand. So it will go on, and just to get to the perfect microbial combination. And at the end, I just like to highlight, like I got some 3-member microbial community, although I am unable to predict what are the actual metabolites that are exchanging between them. Although I have in silico I have predicted some of the metabolic exchange, but maybe some advanced co-culture technique I think that Professor Paul has suggested that could help me to lead to understand like what are the actual metabolites, or maybe 13 CMFA kind of things will help me to understand what are the actual metabolites that are actually leading me to this synergistic metabolic interaction. Those are lead to the degradation of the lignocellulose in a community much better way than in an individual. So thank you for your attention. This is all from my side. And I like to thank the organizer and my PhD supervisor, Dr. Amit Ghosh from IIT Kharagpur, and everybody else associated with this study. So that's all from my side. Thanks.
Thank you, Preetham. We will now have the valedictory session where we'll announce the poster winners. Just 2 other announcements. All participants will receive their certificates of participation only through email. And those participants willing to return your lanyards, please do so to Roshni or Kirti at the reception.
So in the meantime, does anybody want to, I think we can take a few minutes if somebody wants to share any feedback about the conference, especially the younger folks. Don't be shy.
Hello. So the conference was fantastic. I'm actually a phage biologist, and I can actually get a complete different picture of the microbial composition. So I was able to connect a lot with this thing. And I also want to appreciate both Karthik by the way by which they actually organized this conference as being a small meeting that gives enough time for younger faculty members as well as students to basically interact with senior faculty members. So this kind of conferences really gives enough time for us to connect and communicate. So this is fantastically organized, starting from the talks to our accommodation to everything. So thank you very much to Karthik 1 and Karthik 2.
Thanks. And please, and if you have suggestions for improvement and any constructive criticism, it's all the more welcome. Yes.
For the posters set up, it would be the posters from the same sessions were right next to each other. So sometimes when presenting, you might be in the way of somebody else's poster while they're also trying to present. So it was a little crowded. So perhaps next time, the ones that are right next to each other could be from 2 different
sessions. Absolutely.
But
thanks for the
good
point.
overall.
I think Niranjan.
Yeah, so going on the same theme, I found the posters very interesting. I spent like half an hour per poster, and there were so many interesting posters that I've barely seen, like 10 out of the 50 posters out there. Can we have longer sessions, because we seem to be running between talks and posters?
Yes.
I don't know, dinner sessions or whatever.
Yes, and I think you also had a very good point earlier in the day about like a meet the speaker kind of session that would have been like really helpful. Yep.
So almost every speaker. First off, I did enjoy the conference very much. And it was really amazing to have such a small and intimate meeting where everybody could meet each other. So thank you so much for organizing. Almost every speaker, every Flash Talk speaker appreciated Karthik and Karthik. So thank you so much. But I want to give a huge shout out to all the students who led this event. It was really amazing to see, that every small part was taken care of them, right from the pickup to everything. So big shout out to you guys. Thank you so much.
Yep. I'm going to give a shout out to them. Anything else? Okay, I'm not going to, I'm not going to just belabor. So quickly to the poster prizes. I think all of you will agree, especially the poster evaluators, that it was extremely hard to evaluate these posters. We've heard this being said in every conference, but I really totally felt this time because I was on the other side doing all the, Karthik and I did like several rounds of like shuffling to figure out like how do we, sort this, and we said first 3 prizes, then 4, then 5, and then we said and sorry guys, we had to really give short shrift the Flash Talk folks, saying that Flash Talk folks will be separately recognized, right? So you'll anyway get a Flash Talk certificate. So we're going to get you so everybody who gave a Flash Talk can almost assume that they also have a poster prize. So now here are the real, poster prizes for the non-Flash Talk speakers. So the 5 prizes in no real order. And all of these folks are going to get coupons for Taylor and Francis books. So that's our sponsor. So, and thanks to all the participants for really putting in efforts to make wonderful posters, lots of discussions, and as pointed out by Niranjan, we really wanted more time. Yes, I think we have to make this a slightly longer meeting then. So, okay, so with that, Shuchishloka Chakrabarti from IIT Kharagpur, linking the effect of arsenic on methane cycling in arsenic-infested paddy soil. Inderraj Singh from NCBS. Hey, could you just please come and stand here? We'll do a photo because we're going to send you the coupons online. We don't have anything nice to hand over here. Inderraj Singh from NCBS, evolution of transcription factor, DNA binding superfamilies within prokaryotes. Shravani Rajan Rahul from IIT Hyderabad, comparative genome distribution of core essential genes on 2 chromosomes in order vibrio-nels and the evolutionary studies. Lavanya Rajaram from IIT Madras, Cosmos, community and single microbe optimization system. Anish Ganju from the Geo Institute, Mumbai, antibiotic resistance reservoir, stability, transmission and engineering solutions for human health. Okay, so these are the poster prizes. Karthik, if you could come up. Just do a photo. And we'll send you the e-certificates. Everything is e now, so. And maybe we can have one with the Okay, with that, I'm going to really have a short shout out to all the people who were super helpful in running the conference. First, thanks go out to our sponsors. Firstly, Prakash Arunachalam, who's our alumnus, who's given an endowment of $1 million to IBSE, which helps us fund these events without thinking twice. And the Office of Global Engagement, they supported this event hugely financially. I'm also going to acknowledge the specific folks who were there, who ran the show on the ground. Hindustan Unilever, who made a contribution, and also Taylor and Francis for supporting the poster prizes. And then volunteers, please just stand up so that people know your name. For communication, it was Bharati. Speaker accommodation, Nilesh and Yuvaram. Nilesh is here, not here. Still running around, I guess. Participant accommodation, Shraddha, Varsha, Srijit and Sri Krishnan. Transport, Amrish, Saidas, Kanakalakshmi. Venue, Rituja, Shaunak and Arya. I hope I don't have to pack one of these to TA for my class now. Then the sessions, Mazia, Virendra, Sandhya and Prithvi. Stand up. And food, Ashika. Ashika's not here. Outings, Roshan. Roshan's also not here. Registration, Aishwarya, Roshni, Shukriti, Haripriya, Kirtana, Tanisha. Thank you all. Digital, Pratyay, Venkatesh and Ritviz, including all of these things that you see. Poster, Brinda, Varsha, Shabdaini, thanks a lot for running around. I think Aarti was also equally involved. Her name is not here. Okay, and thanks to all the speakers. We had 22 speakers, including 1 virtual. We had 12 international speakers, 10 national speakers, 9 female speakers, 17 session chairs and all. Thank you all so much. And then we had 54 male participants, 56 female participants, 10 faculty, 61 students, 36 researchers, 7 international participants. And I also would like to acknowledge a few other folks at IIT Madras, Professor Ashwin Mahalingam, Dean ACR, who was there for the first day, Professor Raghu, who met with us today as well. And a big shout out to Vinod, Alfred, Pavitra, and Mary. Are you guys there? Vinod is here. Pavitra is there. Alfred is not around. And Sai Shruti is also here. So from OG, they were like a huge support for this. And I know how much they work for other conferences, but because I lead the conference secretariat here, but these guys had a lot more pressure coming and doing it for my own conference, and I really appreciate, they made my job really easy. Thanks a lot to you guys. Anupama from NPTEL, Bhavani, Shanti and Darshini from Code, of course the Vadhwani School of AI for a lot of support, Taramani Guest House, Bose Einstein Guest House, the Council of Wardens, Office of Hostel Management, the Dean Office for a lot of the transport that you had, and John and Suresh for running the e-carts multiple times. And of course, for the cultural programs, I'd like to thank Gyandev, Papu and team. I think you all had a great time. And a few others, Sharmada for design, Sahana Ghosh and Amrita for the conference reporting, Katpaga Travels for travel arrangements, Walker Associates for the hostel amenities, Vaishnavi Events for the poster boards, Kirubai for the banners and audio system, Print Up for all the conference stationery, Jute Palace for the jute bags, Everest & Co for the water bottles, Tanjore Creations, the portrait company for your mementos. I hope you really like them. Nandini Sweets, Eden for the catering. I heard a lot of good comments about the catering. Hope you all like the food. Vignesh for photography, thank you so much. Shukriti for that beautiful rangoli you saw on the first day with the flowers in the front. And Harshita for the banner design. She's no longer here, but she's been helping remotely. Then Shranot at Western Hotel and Raintree. And is Ashmita here? She's still busy. Get Ashmita here, please. She's probably gone off on another of the errands I sent her off on. If she's not here? Okay, so I must mention this that without Ashmita, this whole event would just not have been possible. And if any of you are thanking me and Karthik for the event, I think you should just thank Ashmita for the same, because she's the one who did like 90% of the work. And all that I did was just answer some email messages and WhatsApp messages, trying to get things in shape. So I think a big shout out to Ashmita, who's still very busy doing stuff. So she can watch the YouTube stream later on. Thanks all, and thanks to all the participants. Thanks to everyone out here. It's been wonderful. You see that I never acknowledged Karthik from NPTEL. I see him here. Thanks. I think so. And any other names that were missed, my apologies. I think too many people contributed to making this a great event. Thank you all. Thank you. Okay, thanks all. That's it. So please, join us for tea, and there will be a trip to the IITM Research Park in about like 10-15 minutes once you're done with tea. So it'll be nice if most of you can join for that, especially the international speakers and international participants. It'll be great if you can stay back and attend that to see. It's a very unique sort of setting. With that, happy to call this conference close. Karthik, any remarks? No formalities, so. Okay, thanks. Thank you. This is just a small token.